That was very effective. Welcome, everyone. My name's Siddharthan Chandran. For those of you who don't know me, I'm a neurologist, and I've had the privilege of being in Edinburgh for the last 15 years. And the standout privilege, and in many ways my honor, has been to lead the Euan MacDonald Centre for over that time. So it's a great pleasure indeed to welcome all of you here today, particularly our external guests and invited speakers. You're very welcome indeed. So today and tomorrow is special because what we're wanting to do is, this is a tribute to a life very well lived with unparalleled impact, and that's the life of Euan MacDonald. Many of you in this room would have known Euan, but I'm delighted that Euan's parents are here, Donald and Louise, and also Euan's brother, Angus. I'll speak more about them later in the public lecture in the afternoon. Today's morning session and tomorrow is about research. And it's about research that cuts across boundaries and sectors and geography in the belief and the knowledge that through research you can change lives. And one of the things I picked up from Euan, who I got to know very well from my very first meeting with him in 2008, through to my final meeting with him in summer 2024, last summer, are a few things. Great courage, humor, great humor, love of sport. Hibs was his team, is his team. But the other thing that's particularly relevant to all of us was Euan's unshakable belief in the power of research to transform the future. His belief that tomorrow can be better than yesterday, better than today. He also had the humility and the altruism, like many other patients, I've got to tell you, who I have seen in my own career, Chris would have seen and other neurologists would have seen, who are selfless in that they offer themselves as subjects for study and for research, and they contribute in so many other ways to drive research. So that's the reason we're here, is the belief that through research, we can change lives. We know that through research, through data, you can transform the future. So this event is simply another node in that journey. Many are involved in this. We're focusing on Euan because Euan and his centre and his parents have transformed the landscape for Scotland in this field, without question. Believe me, I could talk about this powerfully for a long time. I won't now, but I might later. So what I'm going to now do is just play a short audio clip which Euan delivered at the opening of NCALS. Those of you who are not familiar with NCALS is the biggest meeting of researchers in the field of motor neuron disease in Europe. And we, as in Edinburgh, had the great privilege to host it in summer 2022, June. This would have been unimaginable 10 years ago, 15 years ago, that 650 of the very best researchers in the world would come to Edinburgh to talk about motor neuron disease. The only reason they were here is because of Euan, directly and indirectly, because of the impact of the Euan MacDonald Centre. There is no doubt about that. That was an extraordinary event, and many of you were here and graced that event with your presence. This speech he gave is powerful, and this speech was also generated through technologies that you'll hear about later, the power of data, speech, AI, to improve the quality of people's lives through speech technologies, one of which has led to one of three of Euan's major legacies, which is called Speak Unique, and you'll hear about that later. Having said all that, technically, I'm just slightly nervous about getting this right. So this is the speech. This is outside, is that Usher Hall? That's outside the museum before the dinner. And that's 600 plus people, and that's Euan.
This is the largest ever, and it is wonderful that you are all together in person and not meeting virtually. Making progress with M&D research is important for a number of reasons. As you all know, it's a particularly brutal, devastating disease in its impact. The nature of the disease and the lack of treatments other than ventilation mean it's a difficult diagnosis for people to come to terms with. The average lifespan for someone with M&D is 2.5 years. I think that should always be borne in mind while discussing ethics and trial design. We need to find ways to reduce the mountains of red tape. M&D isn't a rare disease. It touches thousands of people in the UK every year. There's a cost to society, tying up resources that are scarce, and more importantly, the emotional cost and burden which patients and families bear for the rest of their lives. Clinical trials are crucial to make progress. The huge uptake in participation levels are a sign of how patients value clinical trials. It's not a given that people will put their trust in conventional medicine. Many look to alternative therapies for the simple reason that's where they're given, and in some cases, oversold hope. Patients regularly travel overseas for unregulated phakic stem cell treatment at the cost of tens of thousands of pounds. I'm not scoffing at all alternative therapies because some people find comfort in them. But I do think the lack of treatments available pushes people unnecessarily towards alternatives. So it's important that conventional medicine and clinical trials start to offer real hope. People want to put their faith in you in medical research. That's starting to happen. It's an enormous challenge, but that shouldn't deter you. There are many incredibly smart people who are devoting their lives to M&D research. The more, the better. Please know that it's appreciated hugely. Please keep going, keep collaborating and innovating. And don't forget, we're here and want to help. There's an army of thousands of the most highly motivated people out here. Thanks to all of you. You are making a difference to people's lives with every discovery you make and every PhD student or postdoc you mentor. We'll find the cure one day. Whether it's you yourself or based on the foundations you have laid, I'm convinced we can do it.
What a wonderful speech. That's the ideal way to frame everything we do. I now want to introduce Richard Ribchester, who's going to tell you a little bit about the origins of the Euan MacDonald Centre. For those of you who don't know Richard's now an emeritus professor, which is a very senior honorific title given to the very most senior scientists of our university. So Richard is a neuroscientist, has spent a lot of his research working on the connections between the nerves and the muscle, the NMJ or the synapse. There will be no questions after this talk. I'm keen that all speakers, as best as you can, stick to time. The subsequent research talks, there will be 5 minutes. I'm going to sit at the front to try and help ensure that we stick to time as best as we can. Thank you very much, Richard.
Thank you very much, Siddharthan. It's a great privilege to be here and to give you this little talk about the history of the centre. It's a privilege because it's a very appropriate occasion and to pay tribute to Euan, without whose indomitable spirit, courage, and incredible unremitting resolve, we wouldn't be here today. And thanks especially to Donald and Louise. I can't see them at the moment where they are in the audience at the moment. It's absolutely fantastic to see you here. I thought it would be of interest to those of you who were around at the time when the centre was instituted, and also then for those much younger members of the audience to hear a little bit about how the centre came into being. And so I've entitled this talk a brief history of Euan MacDonald Centre time from beginnings to Big Bang, and say a little bit perhaps about the beyond as well. And I want to say just very quickly something about the conception of the centre, the gestation period, and then the birth of the centre, the Big Bang, the big splash which we then took off with, and some little thoughts maybe about what's been achieved since then and to go beyond. And I try this to make a very swift whistle-stop tour before we get on with the hearing about the real science. So to begin with the conception of the centre, the story kind of starts actually a little bit earlier than this, but back in the autumn of 2005, the Centre for Neuroscience Research, which was the umbrella organisation for many of the researchers in Edinburgh in that time, held one of their annual retreats at the Burn, which is a wonderful location in Aberdeenshire for sitting around and thinking and talking about things. And Peter Brophy, who was chair of the Centre for Neuroscience Research at that time, asked all of the principal investigators there, of whom there were many, to include in their presentations not only their progress in their research, but also ideas for how things might develop in the future for neuroscience within Edinburgh. So this slide is actually from the very talk which I gave at that congregation of neuroscientists there. I had actually, some time before that, been thinking of ways that we might bring together people with an interest in motor neurons and motor neuron disease research. And I had come up with, more or less on the back of an envelope, a list of about a dozen names of people who were in Edinburgh at that time, who at one time or other had done any kind of research involving motor neurons and motor neuron disease. And I presented it as an idea, the possibility of forming some kind of focus group where those individuals might get together and talk about research and talk about ways forward. Well, that went down reasonably well. I think there was some discussion about it. Nothing much happened then until the Christmas lecture, which took place at the Napier campus just down the road in December of 2005. And over the break, Rebecca Devon, who has been working at the Institute of Molecular Medicine on ALS2 variant of motor neuron disease, Caroline Hahn, who had been working on various neuromuscular diseases in horses at the vet school, and Charles Wallow, who was professor of neurology, happened to be chatting over a glass of wine and discovered that we'd all had similar ideas about this notion of bringing together neuroscientists in Edinburgh with interest in MND and motor neuron biology. Well, that glass of wine led to lunch at a restaurant over the road from now no longer with us, but a favourite haunt for popping over to have discussions, scientific and otherwise, with colleagues, the Negociants restaurant over the road from the medical school in Teviot Place. A longer conversation with Charles, with Rebecca and Caroline. That was round about February of 2006. It then fell upon me then to take the next step forward. And the next step forward was an organisation which some of you here will remember, I hope, the so-called Zoo meeting, where I had put together a list of speakers indicated here. The list you can see from the original list had grown to about 15 at this point, with inviting people to bring along their students and so forth. So this was a colloquium of local people sharing their interests and seeing what we might do to get together. Now, I had got wind of the interest of Donald and Louise just about a month or so before that. The organisation of this colloquium was well underway, but it was really Mandy Jackson who told me about the MacDonald family's interest in supporting research in the university. And I wrote a letter to them in April, just before the colloquium at the zoo, inviting them to come along. Unfortunately, I've lost the facsimile of the actual letter, but I have the text here. And as you notice I pointed out that there were a number of us trying to get together, and I invited them along to the symposium. And indeed, Donald and Euan came along, and that was the first time that we met. So that colloquium then led to a meeting. I think it was either the next day or a few days afterwards, where several of us got together, and Donald and Euan came along to that informal meeting to just discuss where we might go from there. And we came up with 5 questions which we figured were appropriate focus for a research group such as they were at that time. These had to do with questions of vulnerability of motor neurons, the induction and spread through contiguous regions, the overlap between MND and other neurodegenerative diseases, the molecular mechanisms, and what the risk factors were, age being the most important one. Well, that then coined or led to the formation of a group which we called EDMOND, the Edinburgh Motor Neuron Disease Research Group, and we had a skyline logo, as you can see there. I put together a website of all the people that were potentially interested in this, and now you can see that the list of those people had grown to about 20 people or so. And we figured that through this further discussion that there was an opportunity here for us to get together and to generate a research program across a broad front of basic translational and clinical research in which various aspects of basic research in terms of cell and molecular biology, genetics, neuroanatomy and physiology, through translational research involving animal and other models, biomarker development, neuropathology, and clinical aspects directly interfacing with patients through clinical trials, psychological interactions, and the development of quality and life aids. There was a real chance for us to develop this. Well, that then led to lots and lots of meetings and discussions with Donald and Euan, documents exchanged. We had many very pleasant, pleasurable, and progressive meetings at the Elephants and Bagels Coffee House, which was over the road from the medical school. Again, now no longer with us. It has some other name now. And that process went on for about a year. And then that gave rise ultimately to what we might call the Big Bang. And this was a splash in the Scotsman newspaper at that time, where Donald and Euan announced their extraordinarily generous donation to establish the centre and to get us launched off the ground as a proper conglomerate of individuals. The search then went on for developing the centre. The remit agreed with Donald and Euan was that we should seek a leader who was a clinician scientist, that we should plug research lacunae in the portfolio of projects that we had, and in particular the omission of iPS-derived motor neuron research, and that we should establish a training program for doctoral and postdoctoral students. The official big splash, though, was a symposium which we had already got in the pipeline before the announcement of the MacDonald's donation, an international symposium where we brought together the individuals in Edinburgh and invited along national and international leaders in the field of motor neuron disease research. Many of those people are here in this audience still today. I think Chris Shaw is here, and Brian Dickey I can see over there, for instance. And we showcased essentially the research that was going on in Edinburgh and heard fantastic presentations from the likes of Don Cleveland from San Diego, Kevin Talbot in Oxford, Jeff Rothstein came over from Baltimore, Chris Shaw, as I mentioned, Pam Shaw, and so on. And here's some photos of that symposium, which was took place at the Royal Society of Edinburgh in the September of 2007. The search for our new leader culminated, cutting a long story short, with Siddharthan, a rather youthful photo of you there, Siddharthan, I think, from that kindly given to me by Rebecca, so blame her, please, for that, and everybody looking cheerful about the moving forward. So that was basically a potted history of how things came about. I just wanted to say a little bit about what, from my own mind, what has been achieved since those early days when the Edmund Group and the centre was set up. The eagle-eyed among you may probably not notice, but in this little paragraph at the back here, I fell into the typical trap of scientists when quizzed by a journalist was how quickly are we going to get anywhere with this, and the stock answer is generally given in 5 to 10 years' time, which is generally considered to be long enough to keep people off your back. But I thought it would be appropriate, actually, to consider where we have come. Just we've come a long way, of course, in very many different areas, but those 5 questions that I laid out earlier on in the talk had a number of implications about what we were trying to find. And I predicted then around about that time that within the next 10 years there would be, for instance, better understanding of genes and environment, risk factors, better understanding of molecular mechanisms, the role of different cell types, the understanding of pathways where protective drugs might be effective, advances in differentiation and engineering of stem cells, advances of understanding growth and regrowth of axons, realistic prospects for a better treatment of the only treatment that existed at that time, riluzole, and improvements in the interface with patients in terms of psychotherapy, physiotherapy, and the development of aids like, as it turned out, Speak Unique. So looking at 10 years from when the centre started, I think we could put ticks in virtually all of those, except at that time in 2017, there still was no realistic prospect of a better treatment than riluzole, I say. But if we step forward to 2025, then with the emergence of the tofersen trial and the success of that, we have seen for the first time, I think, something that's really dramatically effective on admittedly a subset of patients with ALS and with motor neuron disease, but at least with that sets the stage for future developments. And there are ongoing and upcoming developments in the case of FUS, form of ALS, etc. as well. So the challenges to MND and ALS research remain, I think, threefold: to prevent motor neurons dying, to promote compensation by surviving motor neurons, and the big ask is to provide and regenerate lost motor neurons. That's probably too big an ask for the present, but there is encouraging progress in neuroprotection, preventing motor neurons from dying, and for promoting the compensation of surviving motor units to take over the activity of the lost motor units. So finally, none of this would have been possible without the spirit and as I said, the courage and the really incredible resolve and cheerfulness of Euan. My abiding memory of Euan was even in the midst of his disease, and I would say, "How are you feeling, Euan?" And he would beam at me and say, "Very well." Whether he was being ironic or not, I do not know, but it was an incredible experience to have conversations with Euan throughout this process. So wherever the quarks of your consciousness are right now, Euan, then thank you, and thank you also, Donald and Louise, and Angus, who I met for the first time the other day. Thank you all very much.
Thank you very much, Richard. Whilst Rebecca's setting up, I'd like to invite Professor Sabine Liebscher to the stage. Thank you very much for coming from Innsbruck, which isn't quite as tropical as Edinburgh. Oh, you come from Munich. Okay. Sabine is a physician scientist and is going to talk to us about circuits. Circuit biology, circuits in the context of ALS. And you're director of the Institute of Systems Neuroscience at the Medical University of Innsbruck, hence why I thought you were coming from Innsbruck. Thank you very much.
Thank you very much for the invitation to this wonderful meeting. It's absolutely fantastic to be here, also at this gorgeous venue. So I'm very happy, and indeed it's a huge privilege, as Richard just pointed out, to be part of this tribute symposium. I'd like to share some insight into what we're doing in the lab, where we're interested in studying, let's say, the complexity of the brain, for that matter, where we study circuit mechanisms that underlie ALS and FTD pathophysiology. Now, why are we interested in this and believe that it actually represents a really important aspect to ALS pathophysiology that's heavily understudied at the moment? So as you probably know, you're roughly or estimated 86 billion neurons in the brain. They come not only in many, many different flavors, but more importantly, they are connected in very intricate and tightly regulated local and global networks. And it is because of these networks at the end of the day that we talk, we walk, we think, and we feel. And more conversely, disruptions within these networks are essentially at the heart of symptoms typical of a given disorder. And even more so, we and others could show that disruptions within these circuits are not only at the heart of symptoms, but they can also fuel or even trigger the neurodegenerative process per se. And what we found recently is that this is often not only because of the so-called vulnerable cell population, but we also have identified neurons that look perfectly healthy, but yet are changed functionally. And because of this functional change, they drive degeneration of downstream neurons in the circuitry. And so with this in mind, the overarching goal of the work we do in the lab is to really fill this big, well, in that case, it's a white box, but the black box in between the known genetic or molecular external factors, downstream alterations of pathways, and the eventual clinical symptoms by essentially looking into structure, function of individual circuit elements, microcircuits, and global networks. And so the disease per se that we're all addressing today probably doesn't need a lot of introduction to such an expert audience. You all know that this is the most common adult onset motor neuron disease, characterized by the death of upper and lower motor neurons. And we also know that histopathologically, it's characterized by the occurrence of proteinaceous aggregates within cells. They can come again in many different flavors. The vast majority of patients display phospho-TDP aggregates, but you can also, depending on, or more so in seen in familiar cases, see so-called dipeptide repeats, or in FUS mutation carriers, you see mislocalization and aggregation of the protein FUS. And sort of the canonical view on this has been for a long time that the cell develops these aggregates, and then because of these aggregates, you have a lot of downstream, it's essentially triggering a cascade of downstream events that eventually lead to neuronal death. This concept somehow comes with a little caveat such that these aggregates are found in many, many different cells. So not only in the cells that are actually degenerating, so not only motor neurons, and even more so, they're found throughout the brain. So not only in areas that are actually affected. So the question is a bit, how can we reconcile this global proteinopathy with a selective motor neuron death? I mean, we touched upon this, or Richard touched upon this in his introduction already. This sort of concerns the concept of selective vulnerability. But so we, seeing a section like this where you see phospho-TDP across the entire cortical column, for me, the question has always been, what's actually happening? What's happening to neurons that have these aggregates and die? What's happening to neurons that have aggregates but do not die? What's happening to neurons or glia cells, for that matter, that are seemingly not affected by this pathology? So in other words, what's happening to an entire circuit? To address this, we somehow went against the flow of current, and we decided to study the cortical microcircuit. Cortex is characterized in ALS patients by a feature called cortical hyperexcitability. So this is typically measured by means of transcranial magnetic stimulation, where you can then, you put your spindle above motor cortex, and you can measure the potential of the downstream innervated muscle. And in ALS patients, you actually need less of a stimulus, or if you use the same stimulus, you get a stronger response. And this is usually, or the field is sort of spearheaded by colleagues in Australia. And what they've shown in many incarnations of this theme is that cortical hyperexcitability is actually a combination of increased excitation, but at the same time, impaired inhibition. And to some degree, apparently also sort of hyperconnectivity within cortical or cross-cortical areas. Now, so we decided to study that phenomenon, not only because of these interesting aspects, but also because signs of upper motor neuron death is actually a vital aspect to be able to diagnose ALS in a patient. And that still, clinically speaking, represents a bottleneck very often, because to read out upper motor neuron involvement, you still need to have a functional connectivity to the muscle, right? So we also thought it would actually be really helpful to have a better tool for clinicians to be able to readily diagnose or see clear signs of upper motor neuron degeneration. Is it relevant to the disease, or is this just an epiphenomenon? There's actually good evidence that it occurs early in sporadic cases. It can occur prior to symptom onset in familial and mutation carriers, and it does correlate negatively with survival. So I think there's good evidence that the cortex and upper motor neurons per se do play a vital role in the overall pathophysiology of the disease. And so we set out to address this question: what are the underlying cellular and circuit mechanisms that drive cortical hyperexcitability, and how does disruption within this local microcircuit actually affect cortical information processing? Now, just so we're all on the same page here, this is just a very simplified scheme of the primary motor cortex in animals, but believed to be more or less true for humans as well. And you can see in red, these are the upper motor neurons that reside in deeper layers in cortical layer 5B. They receive their main excitatory input from upstream cortical layers in layer 2, 3. And then, of course, there's long-range projections from other cortical or subcortical projections, and within these local microcircuit, activity is regulated by a slew of different interneurons, so of inhibitory neurons. Okay, now, what we did initially back at the time, we decided to use a mouse model to study that, and we opted for the classical mouse model in the field. The SOD transgenic animals that carry a mutation found in the superoxide dismutase gene, known to also occur in SOD patients. These mice, and we can of course debate to what degree mouse models are, an actual replicate of the disease, but these mice have really nice replicates of the clinical, cellular, and the molecular features we also see in ALS patients. And this is just to illustrate that indeed misfolded SOD is accumulating also mainly in deeper layers of the cortex in these animals. to now get an access or an angle to study neuronal function in animals while they actually do something, while they move around, we perform calcium imaging, so in vivo to photon calcium imaging, in animals that are head fixed running on a little styrofoam ball, and depending on the question and experiment we're performing, or ask for that matter, we present them with virtual reality so we can probe motor function, learning, and at the same time record a lot of behavioral parameters to, in the end, be able to interpret what you see here is blinking, right? This blinking sort of is neuronal activity measured by means of fluorescent calcium release. So every time you see this little cell blinking, that means that very neuron was firing. Okay, so this is the overall approach. These are just a few examples of individual neurons and their activity levels shown as so-called calcium traces. And maybe when you watch carefully, you can already see a difference between a wild-type animal and an ALS transgenic mouse. The first thing we did to really try and make sense of all of this blinking, like I said, we actually went for an unbiased approach. So we simply took all neurons in a given field of view and put them into what we call a design matrix. So you can see that every row represents a neuron over time, and we align this with the behavioral parameters we're recording at the same time. We then perform dimensionality reduction and simply ask how much information is essentially covered in that kind of activity in a given field of view. And I hope you can appreciate that it's very high dimensional, meaning if we only look at the first 10 components of the signal, we only cover about 40% of the variability of the activity we see. However, this very first component covers the largest proportion of activity, so we focused on this for the subsequent analysis. And so this first component is mainly driven by locomotion and pupil width. This is not entirely surprising because when we run and when mice run, our pupils will widen, so these parameters are actually interdependent. Now, what we found is that the explained variance to or of locomotion was significantly lower in SOD animals than in their wild-type counterparts. So that meant something was wrong with the information on locomotion in the neuronal activity we looked at. And indeed, when we look or perform a 3D space, so we essentially perform what this is called a manifold representation of neuronal activity. So each data point represents the network state in time. And you see that these network states essentially fall into two clusters. One cluster corresponds to locomotion, and the gray cluster corresponds to quiescence, so when the mouse is just sitting on the ball. In SOD animals, however, these two clusters actually collapsed, right? They merge, they are very close to one another. In other words, the network doesn't really fully represent the state of locomotion anymore. This can also be quantified, obviously, and you see that the distance, the Euclidean distance between these two clusters is lower in SOD animals. If this is true, and the information on locomotion is not well captured by neurons in this network anymore, then if we do the inverse, so that we train a decoder and ask how fast was the mouse running based on the neuronal activity we see, then this performance should also be reduced. And that's indeed what we see here. So we decoded running speed from neuronal activity, and indeed the performance is a lot poorer in SOD animals, confirming our initial finding. So what's going on with the locomotion signal? To address that, we computed so-called locomotion tuning curves. And we did this in early symptomatic animals. So these mice have a bit of a tremor. They still walk fine. They're not severely impaired, but they do show initial signs. And here are a few examples of the neuronal activity we found. And what we did is we simply computed or looked into how well would they respond to running over being active when the mouse is just sitting. And we did it this time within upstream layer 2-3. So this is again the input layer, providing glutamatergic input to our upper motor neurons, and down in layer 5, where the motor neurons reside. And what we found is that in both layers, there is a strong tuning towards locomotion. So unexpectedly, these neurons actually fire a lot to locomotion. We see also that the fraction of running responsive neurons is significantly higher, and these neurons fire a lot more than the wild-type counterparts. So overall, we found neuronal hyperresponsiveness to locomotion in cortex of these SOD animals. This is just to confirm that they're not impaired. So this feature is not driven by changes in locomotion speed, average activity, etc. What's happening if we go to an earlier time point? So when mice are pre-symptomatic at P60, we did the same analysis, and in upstream layer 2-3, again, the input providing neurons, we see the same feature. There's an increase in the fraction of neurons responding to locomotion, and they respond also more vigorously. But surprisingly so, there was no change in layer 5. So upper motor neurons at that pre-symptomatic stage are functionally, at least for this parameter, not impaired yet. So the initial driver actually comes from an upstream population. And the model we came up with then is that if we have these guys, the layer 2-3 pyramidal cells, that turn hyperresponsive, so it's just another feature of hyperexcitability, if you wish, and therefore provide excess glutamatergic stimulation of the downstream upper motor neurons, such that the disease process would actually lower or propagate across the microcircuit, across cortical layers. If this is true, can we interfere with that process? Can we target that layer 2-3 population, sort of inhibit it, and therefore prevent the propagation of the pathophysiology? To do so, we used a chemogenetic approach. So we just expressed a design receptor in these upstream layer 2-3 cortical layers. We stimulated it using CNO over a month in the pre-symptomatic stage. And indeed, these mice, they gain less weight. Their symptom onset is delayed. And even at late stages, a month after we stopped the treatment, they still perform better in a motor test. So having found the role or the critical involvement of these, critical and early involvement of these layer 2-3 pyramidal cells, we wondered, this is the SOT mouse model, is this something specific to this mice? But of course, we had actually published a couple of years earlier that we find similar features in another mouse model that's expressing a mutant form of FUS. So also here we found an increase in excitability in these upstream layer 2-3 cells. Now, what about humans? Obviously, we can't do these experiments in humans. So what we decided to do is we opted for a single nuc RNA-seq approach in the hope to find features that would allow us to find also changes in these cortical layer 2-3 cells. And sure enough, if we just look at excitatory neurons in these patients, so these were C9 familial cases, we see a significant reduction in a cluster, in the biggest cluster called cluster 0 here. And when we look at the features of this cluster, these actually correspond to intratail-encephalic layer 2-3 pyramidal cells. So this is more or less the same population that we also found to be dysfunctional in the mouse models. And when we quantify this is just a density plot to show you that really exactly this cluster 0 is lost. Here the quantification, we validated this using RNA-scope. So by having identified these two long non-coding RNAs that are specific or that classify this cluster, we can also in the human tissue show that not only is there a massive drop in excitatory neurons in layer 2-3, but it's specifically this particular cluster we identified that is lost almost completely. All right, so what's going on with these layer 2-3 cells? Why are they hyperexcitable? Why are they vulnerable? Although obviously we must have overlooked this in the past, they're not as prominent as beta cells, so it's probably easy to miss that. And why is it locomotion dependent? Why would we only see this when the mouse is running? And so we wondered, how to approach this, and we do this now from very different angles, obviously. But one thing that was striking is that we know from the literature that locomotion does not only hinge on glutamatergic input, but it is also driven by neuromodulators. And first and foremost, driven by noradrenaline, released by neurons that reside within the locus ceruleus. And so we also have a project where we look into astrocytes, which have been proposed to play a role in the disease. And when we did this, we already found that astrocytic calcium signals are reduced, and also astrocytes are driven mainly by noradrenaline. So what we did here is to simply measure in a moving animal the release of noradrenaline in cortex using a genetically encoded sensor for norepinephrine or noradrenaline. And I hope you can see that with locomotion, you get this increase in fluorescence, which we can quantify, and which was almost absent in the SOD animals. We did the same in FOS, where we also see a massive reduction in NE release. And while NE release typically scales with locomotion, so the faster you run, you know, when you do your morning jog, the more noradrenaline is actually released in your brain. And this scaling effect is also almost completely abolished in these animals. So is this true in tissue? And so here we teamed up with Caroline Rouault in Strasbourg, and she actually measured a complete set of neurotransmitters, neuromodulators, and she, just like we, using our imaging tools, found a almost specific and selective reduction of noradrenaline in the tissue. It's not glutamate, it's not GABA. Serotonin has been described before, a tiny bit, but there was a huge, huge reduction in noradrenaline in the tissue. So why is that? In animals, what we did is to quantify the number of somata in the locus ceruleus to just check whether there's actually neurodegeneration ongoing, but that wasn't really the case. There's an early change, only in mice that are roughly a month old. What we found, however, is that the innervation of cortex is massively reduced. So it seems that the noradrenergic fibers don't really see the target properly in cortex. So this is still a matter of ongoing investigation at the moment, but since this is happening so early, one might even speculate whether there is a neurodevelopmental aspect to this feature. What about humans? We also obtained human tissue, postmortem tissue, and again confirmed this reduction in noradrenaline both in motor cortex as well as in the locus directly, not so much in spinal cord, but there was not a lot of noradrenaline in the spinal cord to start off with. Can we now link this back to the hyperexcitability that we've seen? And to this end, we decided to use a parameter from EEG recordings, or in mice ECOG recordings, which is called phase amplitude coupling. And this was spearheaded by my dear colleague Caroline as well, so she had that idea. And we computed this parameter, and indeed, when you look into the interaction between the slower frequencies of theta with a higher frequency, in that case gamma, you can see that the amplitude of the higher is sort of entrained by the frequency of the slower bed. So this is called phase amplitude coupling, yes. And so this matter, or this feature, was also massively impaired in mice, as you can see here, in 2 models. And then we teamed up with Véronique Marchand-Pouvert in Paris, and she did the same measure in ALS patients. And also there we found a significant impairment of this phase amplitude coupling, primarily over the electrode above the motor cortex, and importantly, over motor cortex, the impairment of phase amplitude coupling scaled negatively with the disease severity. In other words, the more severe your phase amplitude coupling is impaired, the faster the disease progresses. Okay. So then we went back to animals to link it to NE. And what we did simply is mimic disease by using a toxin, DSP4, that deletes or reduces axons in cortex. So we mimic the deficit in adrenergic innervation. And indeed, when we measure phase amplitude coupling, these otherwise wild-type animals show the deficit. Can we do the inverse and rescue disease mouse models? And here we injected a precursor of noradrenaline. And then when we measure phase amplitude coupling again, you do see, while in wild-type animals it doesn't do much, it significantly improves phase amplitude coupling. So there is at least a partial rescue. Okay, with this, let me quickly summarize what I was trying to convey today, is that we believe that cortical hyperexcitability is likely caused by circuit level deficits. These include layer 2-3 cells that become hyperexcitable prior to upper motor neuron dysfunction. They provide with this axis glutamatergic stimulation of upper motor neurons. They also undergo molecular changes and likely degeneration in patients. And the cortical hyperexcitability per se seems to involve a deficit of noradrenaline And for this reason, we believe that neuromodulators can actually represent an alternative therapeutic target for the future. Now, this was a massive collaborative effort. First and foremost, I'm really indebted to people in my lab. The first part was spearheaded by Vanessa Khan, who is now at Stanford, then Evgeny, Liliana, Sheni, Chandran, and Zeynep contributed to this. A big thanks goes to my amazing colleague Caroline Rouault and Luc Dupuis, both at INSERM in Strasbourg, Smita, who is now in the US, and Veronique, who did the recordings in patients. And with this, I'm done, and I'm happy to take questions. Thank you very much.
Thank you very much. That was great. Are you based in Munich or Innsbruck?
Officially in Innsbruck, I'm still commuting.
I see. I'm sure there will be questions. I have a couple, but a question from people? Whilst people are thinking, could we comment, a lot of the animal work is in the SOD model.
Yes.
And then you show the FAST. In terms of replicating that in TDP models, can you comment on that?
So this is sort of ongoing. We're trying to get the permits. You know, animal work is slow, and yes,
the complexity of experiments.
Yeah. So TDP is sort of next. I mean, there is good evidence that C9 models show similar features in terms of cortical hyperexcitability. Whether that refers to locomotion-dependent hyper-responsiveness, I can't tell at the moment. But it could be.
There's a lot of work now in terms of 3D human models. Including cortical. Do you think there's any role in those systems for trying to get at some of the questions you are?
You're referring to organoids?
Yes.
Yeah.
You can say no.
I am inclined to say no.
Okay.
For several reasons. I think as far as so I'm not an expert, so I might be completely off and wrong. But so I've been told that the center is a bit necrotic. I doubt whether the circuits form the way they do. Many of these lack certain glial components. You can add them to the system. But most importantly, for at least that part of our work, I'm not aware that any of the cellular models, be it IPCs, be it co-cultures, be it organoids, actually has or sees neuromodulatory input.
Okay. I think that's a very important observation, something people could discuss. I've always told Bhuvanesh he's overrating stem cells and 3D systems, and you've just confirmed that. Brian Dickey.
Thanks, that was a great presentation. There is a school of thought that the hyperexcitability may actually be driven by a lack of inhibitory input. So ALS is actually an interneuronopathy. How do your findings square with that school of thought?
No, I think they perfectly align with that idea. So we still haven't figured out why these layer 2-3 cells become hyper-responsive. And the lack of internal, and we've actually shown this nicely for the FAST mouse model, where we see mainly a lack or a loss of inhibitory synapses. We've now done, but this is work that we do with Luc, so I don't feel comfortable sharing all of this because it's actually about to be submitted. There is good evidence for interneuronal involvement, and it could to some degree also be involved in the feature we're seeing here. Whether or not this is at the end of the day all downstream of neuromodulatory deficits, I don't know, because all of these neurons have adrenergic receptors in many different flavors. And it could be if you reduce adrenergic input, you primarily hit interneurons, and because of that, you sort of unleash excitatory neurons, you know what I mean? So it's all interconnected, yes.
Thank you. If I could just ask you a last question. What do you think, Sabine, are the clinical implications if this all stacks up? What is modifiable?
Yeah, so we've started to look into adrenergic modulation, which is easy because we have a lot of FDA approved or generally approved drugs. The question for us is at the moment whether that is only a very early feature that is not a target at late stages anymore. We've done, or Caroline has done a treatment, and we've done the imaging for this with yohimbine, sort of to inhibit the autoinhibition. And then we rescue, and it's actually part of the paper, we rescue phase amplitude coupling for about 3 weeks. And then the network sort of goes back. And I think this is something to bear in mind for any treatment, any neurodegenerative treatment. If we interfere with these systems, the system adapts, right? This is also true for Parkinson's, etc. So maybe we might have to come up with treatment regimes where we intersperse a washout period to prevent the system from adapting to it, right? So this is sort of what we're going, the directions in which we're going. I think another promising approach could be selective noradrenergic uptake inhibitors, which should have been, or, you know, there was one paper where they treated ALS patients, but because of depressive symptoms. But then in that paper, they didn't report the motor symptoms, right? It was more like, so this is doable and clinically, I think, really straightforward. It's too early to tell.
We should talk about this later.
Okay.
That's great. On behalf of us all, thank you very much. Thank you. Next speaker is Tom Gillingwater, who many of you will know. Tom is the Professor of Anatomy here in Edinburgh. This is a very prestigious post. I hadn't appreciated you're the 15th only, which tells me that people of Professor of Anatomy must live very long. In addition to your role as the head of anatomy, I know you do a lot of pioneering teaching online and in person, including this terrific partnership with the Royal College of Surgeons of Edinburgh. But you're here because of your research around motor neuron disease, synapses, also to do with SMA, which I'm sure we'll hear about. But it also gives me an opportunity to thank you for what you've done for the Euan MacDonald Centre from the very beginning. You've been one of the absolute bedrocks of the centre. So we're delighted for you to speak. Thank you.
Thank you, Siddharthan. That's an incredibly generous introduction for me. I'd like to thank Siddharthan and Rebecca and all the Euan MacDonald Centre team for putting together this symposium and to give us all the opportunity to really remember and celebrate Euan's life and contribution to motor neuron disease research. So I hope you'll forgive me if I take the first couple of minutes to tell you a short anecdote rather than get stuck into some science. I first met Euan at the famous or infamous Zoom meeting that Richard referred to, and I was really struck by him. There was a chap who was the same age as me. We were born a couple of years apart, who, to be honest, I was in awe of. His strength, his willingness to be open and to talk to myself and others in the community was really quite incredible. And I left that meeting feeling very moved and motivated. I just think, okay, this is something that I know Richard and Siddharthan have been pushing forward, but people like Euan really were driving forward our motivation to do this work. And we've heard about the really big scale differences that Euan, Donald, family and friends have all made to the Scottish and global ecosystem in MND research. But I'd like to tell you about how he made a difference in a much more small but equally impactful way. Euan had asked if he could have a chat with me. It must have been about maybe 2009, 2010. If you could come and see me in anatomy in the old medical school for a chat about early career researchers. And that was something that Euan cared a lot about. He wanted the young next generation of MND researchers to be supported. And he always asked, how's the PhD students getting on? How are the postdocs getting on? So we arranged a meeting and he came to see me. And a couple of days before that, we'd had some bad news in the lab. One of our experiments hadn't worked. Now, I'm sure everyone sitting out there knows that feeling very, very well. But I was feeling pretty low. And Euan came to see me, and I was very aware of how important he was in funding and supporting a lot of MND research. So I thought, I'll hide that from him, and I'll put a smile on, and we'll have a chat about early career researchers. And he came in, and those of you who know Euan will know he had a twinkle in his eye, always. A twinkle in his eye that was special. And he sat down and we were going through pleasantries and he said, what's wrong? Do I tell him or no, I'll tell him. The experiment didn't work. And I was sitting there thinking, he's going to chew me out and tell me how terrible we are and how we haven't tried hard enough or whatever. He just looked at me with a smile said, shit happens, try again. And we moved on to talk about ECR stuff. I'm sure he wouldn't have given it a second thought that that was what he said to me. But that try again absolutely changed my mind and made me feel so much better. And then we went back to the lab and we did try again. And what I'm going to present to you now is a result of that trying again, where those probably 3 seconds of my life and my time with Euan absolutely made a difference. So hopefully we can celebrate the small differences as well as the big things that Euan did. So what did he help try and stimulate? Well, my lab for many years has been interested in trying to answer perhaps one of the questions that Richard highlighted in his initial presentation to us, which is, can we stop motor neurons from breaking down? Quite a simple kind of target, you'd think, in many ways, but as we all know, when you try and actually get to the bottom of doing that, it's much harder than it sounds. And so ultimately, the kind of goal in the lab for many years has been, can we develop the pill, the treatment that we could give to people living with motor neuron disease conditions? And I'm being quite broad in that definition, because we at the time were working largely on a childhood form of motor neuron disease, spinal muscular atrophy, but also adult onset ALS or whatever. And can we simply just create the treatment that will prevent the motor neurons from breaking down? And at the time, there were several ways that we thought that might be done, or ways we could actually try and address some of the challenge. And it was maybe target the disease cause, go after genetic causes of the disease, and that's something that we've already heard about, Tofersen and other things that are coming through. Do we target independent modifying factors, things that aren't related to the disease cause at all, but that might be able to influence how motor neurons behave and how they live and how they die? Or maybe we can try and repair the damaged tissues. And my lab was really looking at the middle of those three. We thought, could we try and use our knowledge and maybe the different models we had available to us in the lab of different forms of motor neuron disease to say, are there perhaps a commonality of independent modifying factors that we could identify and then target therapeutically to try and make motor neurons a little bit better, to try and help them when they're undergoing kind of stressful disease trigger. And so, as an anatomist, we're quite simple of mind. We like to start with simple things by looking at stuff. So one of the first things you do when you look at motor neurons in motor neuron diseases, and again, I'm using that term purposefully as a broad term, is that not all motor neurons are the same. And in the disease context, not all motor neurons die in the disease. And that still, I think, is a bit of a head scratcher. We don't really understand why that's the case. But here's an example from the work we did in our lab with a mouse model of ALS, whereby if we looked at one of the earliest neuropathological events, of which motor neurons, lower motor neurons are involved, which is disruption of the neuromuscular junction, we can see some motor neurons are obliterated in the disease, and yet other motor neurons are comparatively spared. And rather bizarrely, from an anatomical perspective, those motor neurons could sometimes be coexisting side by side in the body. So it's not that only some bits or regions are affected and others aren't. So there must be something fundamentally about the intrinsic properties of some motor neurons that change how they respond or react in the disease. So that's work from a mouse model of ALS. But at the time, when Euan was really kind of picking me back up and getting me to go again, we were actually doing something similar in a mouse model of spinal muscular atrophy. And again, it's exactly the same kind of question of, do all motor neurons react or die in the same way? And lo and behold, no, they don't. There are some motor neuron pools that are very vulnerable to the disease and others that are seemingly resistant. So that fundamental kind of anatomical observation gives us an opportunity to then isolate or pinpoint different pools of motor neurons and ask what might be the underlying cause of that. What might be the molecular features or aspects of some motor neurons that make them more resilient or resistant to disease than others. So this is where we performed some work in healthy wild-type mice. This is not diseased mice, this is healthy wild-type mice, where we could retrogradely label individual pools of motor neurons that we know were differentially vulnerable in disease. And we did this in the hind limbs. I don't know if I can use my pointer, it's not going to work, no. The bottom left of the slide, so these are three muscles of the mouse hind limb, all sitting very close to each other. The motor neuron pools are very close to each other in the spinal cord, so we're trying to keep things kind of like to like. We could inject a tracer into those different muscles, label their motor neuron pools back in the spinal cord, and then cut them out with laser capture microdissection. And this shows how old it was at the time. We did RNA extraction and then microarray. Remember that? Microarray technology to do transcriptome profiling. And the graph on the right basically shows changes in the expression levels of a group of genes, I'll tell you what they all conform to in a minute, who all showed a very similar pattern of expression. So on the left of the graph is expression in resistant motor neuron pools, motor neurons that were not breaking down in the disease. In the middle is intermediate motor neuron pools that were kind of resistant, kind of vulnerable. And then on the far right, the vulnerable motor neuron pool innervating the tibialis anterior muscle. And they all trended the same way. They reduced their expression as you progressively made the motor neuron more vulnerable. All of those genes, transcripts on that graph, are involved in bioenergetics pathways. They're involved in the handling and production of energy within neurons. And so far, to date, we've managed to look at one of them, the one in red. Now, there might be many other very important ones in those black lines we haven't looked at yet. We just haven't had time to do it. But one we did pick out was PGK1. And what you can see is this is a gene that's strongly expressed in resistant or preserved motor neuron pools, and it's got much lower levels, basal levels, not in disease context, basal levels in motor neurons that are going to be vulnerable in the disease. And then, to cut a long story short, we went on to show that this gene, this enzyme, phosphoglycerate kinase 1, or PGK1, a key enzyme involved in glycolysis, is strongly expressed in motor neurons, and it has increased expression in these disease-resistant motor neurons. And we can see that in vivo, we can replicate it in vitro if we do cultures of different neuron populations, but basically it's a strongly expressed neuronal protein. Now, we did a fair amount of work, I'm going to gloss over here, but we ultimately came to the conclusion that if we boosted PGK1 in all motor neurons, we could perhaps convert all motor neurons to being of the non-vulnerable, the disease-resistant type, i.e. we could try and protect neurons by giving them an energy boost by activating or giving more PGK1. We've done that in a range of different model systems, but I'd like to just show you an example of an experiment, in this case from zebrafish. And this was something that came from a Euan MacDonald Centre meeting where I was a devotee of mouse research, and Katerina Becker tapped me on the shoulder and said, "Don't be stupid, use zebrafish." And I laughed and chuckled and went away thinking, no chance. Now we use zebrafish all the time. They're an incredibly powerful model. So these are just some fish models of, in this case, an uninjected control on the left. And if I hopefully click the thing, the video will play. You can see we tickle the tail of the test. There's a much more technical term for it, but it's the tail tickle test. And you tickle the zebrafish and it swims away. It doesn't like it. Now, if we produce a zebrafish model essentially of TAR-DP mutation, so a TDP-43-like model, and I'm going to start the video, we tickle the tail, there's an initial response, but you have to trust me, that video is still playing, that fish isn't continuing to swim away. So it's lost the kind of neuromuscular function we associate so much with motor neuron disease. Now, if we take those fish and we inject PGK1 RNA, so a genetic boost of the PGK1 gene, if I now click play, we tickle the fish and it resumes the neuromuscular ability to swim away. So we think this was some evidence that actually boosting PGK1, I'm just going to go through those videos, was beneficial. I'll just show you some actual data to confirm that I've not picked on an anecdotal nice example of one fish that fit our hypothesis. Basically, if you boosted PGK1 genetically, we could improve neuromuscular function. And if we did the morphology, we could protect motor neurons. And we've done this in SMA models. We've also done this in ALS models. So that's all well and good. You know, injecting RNA into zebrafish is great, but it's not something that you can take into the clinic. So the question was, is there a more therapeutically amenable way of targeting PGK1 that would allow us to do the same kind of thing, but in a way that could be taken into patients? So is PGK1 druggable? The short answer to that is yes, it is. We benefit massively from this very large field at the minute of repurposing, whereby people are identifying alternative functions of well-known and approved drugs. And in our case, there was a paper published in 2015 by Chen and colleagues which showed that a very widely prescribed drug for high blood pressure and enlarged prostate called terazosin or terazosin, depending upon your pronunciation, has a side effect of also activating PGK1. So it's not the primary function of the drug, it's not what it was designed for, but that's something that it actually does. It happens to also boost and activate PGK1. Now, importantly, on the right side of the slide here, you can see the UK NICE information, which basically tells you that terazosin is safe and it's widely prescribed, and it's something that can be used in patients without too much worry about whether it's going to do damage or any harm. So we started exploring, as you would, whether or not this repurposed molecule, which can be bought for kind of 10p a tablet rather than crazy money that many modern technologies require, would have any effect. And the first evidence we got was back in a spinal muscular atrophy zebrafish model, and that's the slide, or the images on the left there, whereby we could actually protect motor neurons simply by giving them terazosin. So in the top panel, the control panel, those little green streaks growing down are examples of individual motor axons from motor neurons in a healthy zebrafish. The middle panel is in an SMA model zebrafish. You can see that those axons are not growing out properly and they're aberrantly branched. And in the bottom panel, that's a spinal muscular atrophy zebrafish where we've given it terazosin and we massively improved the motor neuron health. Just to show you again some data that kind of backs it up in an ALS context. So we did the same thing in an ALS context, but this is now using two different zebrafish models of ALS. So we used a TDP-43 approach and we used a C9orf72 approach, so two different genetic causes of the disease, and we could find something similar, i.e. giving the fish terazosin improved their motor neuron health in vivo. So this was kind of encouraging. I would reiterate terazosin is not a drug designed to be a PGK1 binder and activator. We're kind of taking advantage of a kind of secondary nature of the drug, but it was enough to encourage to think it could be a useful, at least lead molecule to get us quickly towards the clinic. But you need to do a bit more than zebrafish work to convince regulators and colleagues that that's worth doing. So we moved into a Thy1, so this is a neuronal specific driven TDP-43 mouse model. This is a very aggressive mouse model of disease that we use in our lab. So the mice get sick a few days after birth, and then they're dead normally within about 3 weeks after birth. So very rapid, very aggressive disease, very difficult to rescue as a result compared to the more long-lived, long-lasting models. But even if we gave terazosin to the Thy1 HTDP-43 mouse model, we could see significant improvements. And in particular, I'd highlight the graph on the top right of panel B, which is the clinical score. So we use a clinical scoring system in our lab, developed by Helena Chaytow, an excellent senior postdoc who does a lot of this preclinical work. And she's developed a scoring system that we can use multiple readouts to check how healthy or otherwise from a neuromuscular context these mice are. A score of 3 is a mouse that's basically, we would have to be culling it for humane reasons for our home office license. A score of 0 is essentially otherwise healthy. And you can see quite a significant drop in the clinical score of the mice that we treated with terazosin. So this was really something that we felt was a positive step forward, and we've done a lot of this preclinical work in collaboration with long-standing collaborators at the University of Oxford, headed up by Kevin Talbot. And Kevin and colleagues at the time were encouraged to undertake what we'd probably call an experimental medicine study. So this is a small-scale study in patients. It's not a large clinical trial. It's not kind of MND SMART level, but it's designed to try and identify whether there's a signal with the drug, with the intervention, that perhaps would encourage you to take it forward into clinical trials. So Kevin, Alex Thompson, Martin Turner and colleagues set up the terazosin repurposing study in ALS, or TRUST trial, run at the University of Oxford. And we finished enrolling all patients earlier this year, and we were due to report. Kevin is going to be presenting results from that in November of this year. So we've managed to go from an initial discussion in the lab about what might be a way of targeting PGK1 to a clinical trial in the space of about 6 or 7 years, which I think given the timescale of these things normally was really quite quick. And in addition, in parallel, in the States, a group being led by Kumar on the right side here have been working for many years in parallel to what we've been doing on PGK1 and terazosin in other neurological conditions. They're particularly interested in Parkinson's disease. And so for those of us, and I count myself as one of them, who believe there's an awful lot of overlap and potential therapeutic overlap between different neurological conditions, the fact that they found potential benefits of targeting these pathways in this protein and using the same drug, terazosin, has given us a lot of encouragement. They've been pushing forward with a lot of clinical trials in the States, and they've just announced it will be on clinical trials websites later this month that they are now also doing an ALS trial with terazosin, and they are actually using a modified protocol to what we did in the TRUST trial. So hopefully that will give additional evidence either way for whether or not this is a molecule we can take forward. But just to finish up my talk, I want a couple of slides to show how this, from a basic research perspective, isn't actually the end. In many ways, you might think we've handed it over to the clinicians, they can do their bit now, we sit back, put our feet up and kind of move on to the next thing. But actually, one of the things we built into the study design when working with Kevin and colleagues was a cyclical study design where clinical data and understanding was fed back into our research lab. And what that meant was we were empowered to be actually still exploring things like mechanism of action and trying to find ways of optimizing and improving treatments whilst clinical studies were going on. And so one of the questions we fundamentally asked was, well, what does terazosin correct at the molecular level? We've seen that it makes things better, but is it doing what we think it should be doing and targeting bioenergetics, or is it doing something completely different? So this is just a summary of data, again, from Helena's work in the lab, where we did a proteomic screen, an unbiased proteomic screen of our TDP-43 mouse model, the ALS mouse model, and we simply asked what happens to protein levels in the diseased mice compared to controls, and then what happens to those same proteins if we treat with terazosin. So the two heat maps here just show expression on the left of upregulated proteins and on the right downregulated proteins, where the first two columns show a wild-type mouse compared to the diseased mouse. And what you can see in the left panel is at the middle column, at the top of it, it's bright green. These are proteins that are upregulated in the disease. On the right-hand side, the downregulated proteins, the middle column at the bottom of it is bright red. Those are proteins that are downregulated in the disease. But if you look at the third column in both of those, this is where terazosin treatment's been added, we've almost completely corrected those proteins back to what it should look like in wild type. And if you ask the question of, well, what are those proteins? What are the proteins that we've corrected or improved in the model? The panel on the right shows us a kind of pathway analysis. They are largely around bioenergetics and neurodegeneration. So I think that strongly encourages us to believe that it's doing what it should be doing, what we hoped it would be doing. It's actually targeting those pathways and improving them and correcting them, at least in this disease model. But a final thing to show you is that one thing we'll hear about a lot more over the coming afternoon, or morning, afternoon, and tomorrow as well, is that we know that it's not just motor neurons that are playing a role in ALS, and yet we discovered this protein, PGK1, largely in a screen of motor neurons, but we asked, well, could it be doing anything else? Could it be targeting other cells and disease processes in ALS? So we know that in our mouse model, again, the TDP-43 mouse model, we have a lot of gliosis. We have reaction of glial cells to the disease process. So we have increased level of GFAP expression, so astrocytic activation, and IBA1 labeling of microglia activation. So we know in this disease model that gliosis, or aberrant activation of glial cells, is a major phenotype. And if we look at both GFAP and IBA1 signaling, so astrocytes and microglia, both of those are markedly reduced when we treat with terazosin. So it's doing something more than just activating or boosting bioenergetics just within motor neurons. It's doing something we think probably at a more systemic level as well. So just to wrap things up there, hopefully what I've shown you is that disease-independent modifiers can be identified. We can find kind of molecular features of motor neurons that do impact on how they respond in disease, and PGK1 is perhaps just one of those examples. I would never make the case it's the only one or the most important. There are probably many more to be discovered, probably many more powerful ones yet to be discovered. But I think it encourages us to keep moving down that avenue and try and find some of those modulators. Increasing or activating PGK1 can ameliorate motor neuron pathology across different forms of motor neuron disease. It's not specific to one form, one genetic subtype. It seems to have quite a broad potential as a therapeutic approach. And so we now have clinical trials examining this by repurposing terazosin in ALS patients, but perhaps excitingly as well, there are efforts to try and identify specific PGK1 activating drugs, not relying on a repurposed drug or something that's kind of an off-target effect, but something more specific to see if that can have any benefits as well. And hopefully what I've shown you as well is that this kind of circular study design is a really powerful way of keeping clinical and preclinical groups interacting, working with each other, rather than doing anything in a parallel line where one group stops and the next group starts. So this slide is woefully inadequate. I realized that, and as I started trying to think of every person I should name, I realized I couldn't. So there are many, many colleagues we've worked with over the years that have been a key part in this story, and I thank all of them. But perhaps I'd just like to finish by coming back to Euan. And just a reminder that it was Euan telling us to try again, go again, shit happens, try again, that really allowed us to drive this project and others in the lab forward. And so for that, I hope we've repaid him at least in part for his trust and his faith in us. And I hope this just gives an illustration of how Euan really influenced MND research in Scotland at a global level, but also just individually to me personally and to our lab. Thanks ever so much.
That was terrific, Tom. There will be questions. Yes, I can see that. We'll start with Professor Shaw.
Thank you. That was a wonderful talk. I'm just going to deviate slightly. I mean, the energetic story is very powerful. Obviously, the proteomic survey accounts for that. But this drug also increases HSP90, and heat-shock proteins are potent means of removing aggregated proteins. And I wonder whether you'd explored that aspect and whether you saw terazosin actually decreases TB43 burden in the mice.
Great questions. We haven't looked. No, we haven't looked. It's absolutely an area that you would predict would be something to explore. I don't have any data I can share on that.
Great talk, thank you. Did I get it correctly that you started treatment? I mean, in the fish, it was sort of a genetic treatment anyway, right? Or did you? And in the animals, I assume you also gave the drug super early before they actually develop any symptoms. So, what do you think is happening if you were to start treatment after there is degeneration, there is symptoms? You know, would you think it would still be beneficial then?
That's a fantastic question. Yeah. So, to be absolutely clear, you're absolutely right. So all the treatment that I've shown you here is essentially delivered before, or at least in the very earliest stages of any disease onset. Unfortunately, in our HTDP-43 mouse model and in the zebrafish models, they're not long lived enough to do an experiment where you would perhaps do a later initiation of treatment to see if you could still have any benefit, which is going to be more akin to what you're going to have in a patient population. We do now have a much longer lived ALS mouse model in the lab. Again, Kevin Talbot's generated a TDP-based mouse model, and so those are the kind of questions that we hope to answer. But at the minute, I couldn't tell you what would happen if you delayed onset of treatment.
Can I get a second one? Sorry, the terazosin, the proteomics data, it almost looked as if it was overcorrecting, right? So here comes the question: is this a recreational drug for wild-type mice and humans as well? You know,
Yeah, who knows? I mean, yeah, that's quite striking. And actually, if you look at some of the behavioural readouts as well, if you look carefully, the ALS treated animals are better than the controls. So, is it doing something? I don't know. One thing I would say about terazosin, this is perhaps just to try and caveat it a little bit, is that it does have quite significant side effects. I mean, any drug that's going to be targeting blood pressure and the like isn't the best kind of treatment that's going to be improving your general health in some way. So it's not some super drug that's going to be, we can all be taking and we'll all feel great from it. But yeah, it's an interesting observation. I don't have an answer for it, but it's something we're seeing repeatedly.
Thank you. Because of time, I'm going to stop here. Thank you very much, Tom. Thank you, Sabine. Thank you, Richard, for the opening session. We're going to start here at 11:45, not 11:40, just to give people time, but 11:45 sharp, and Gareth Miles will run this very tightly from then. Thank you.
Okay, hopefully you can hear me okay. Put that up a bit higher. All right, welcome back everyone. We'll get the next session going. So first of all, just briefly, my name's Gareth Miles. I'm going to chair this session. I just wanted to mention very briefly some sort of recollections of my own working with Euan and the Euan MacDonald Centre. So I was lucky enough to arrive in Scotland in 2007 and joined St Andrews University at that point. And luckily enough, as you've heard from Richard already, that was about when the Euan MacDonald Centre was being established. And actually for me, it was Euan who was one of the key folk who somehow was aware I was coming to Scotland and had mentioned it both to me and members of the Euan MacDonald Centre in Edinburgh, and that really led to this connection. And I think I might have been one of the first outside of St Andrews, rather outside of Edinburgh, sorry, to join the Euan MacDonald Centre, and that really led to a fantastic series of collaborations, particularly with Siddharthan and his group over the years, and Bhuvanesh and others. So really grateful for that. I remember chatting to Euan, who, much like Tom, similar age to me, and he'd just been to New Zealand. You may be able to tell that I've got no accent because I'm from New Zealand, unlike the rest of you. So I really enjoyed talking to Euan about New Zealand, about rugby, and about St Andrews, which is actually where he'd done his degree. So fantastic chap, and for me, he also, as Tom described, sort of gave me a boost to get going in this work and to work with others, particularly across Scotland. So it's a real pleasure to be chairing this session where we have some key folk from our community, some former, although maybe never a former member of the Euan MacDonald Centre, but former members of the community and current members of the community. And I think it's a really nice demonstration of the community that's been built through the Euan MacDonald Centre. I think it's very collaborative. As I say, extends well beyond Edinburgh, and I think some fantastic science has occurred through those collaborations, and I'm sure it will continue to grow. So the first speaker that I'm going to welcome up is Matthew Livesey. Matt's currently a senior lecturer at the Sheffield Institute for Translational Neuroscience. Matt was formerly at Edinburgh, and as someone who collaborated with Matt back in the day, it's a real pleasure to invite him back. So welcome, Matt.
All right. So it's a great privilege to be back. It feels like I've come back home. So importantly, one of my great memories here in Edinburgh was having focus groups with Siddharthan, Euan, Donald, Bhuvanesh, Karen, and David Story, often late into the evening where we'd talk about our projects. And of course, we'd have to talk about our projects in a lay scientific way so that they'd be understood. And I think importantly, what those meetings did was install a bit of drive in me, which was definitely needed. And importantly, what was needed from myself was to make the disease a personal thing. And that's what Euan has done for me. So this is a little bit bold. It's all unpublished. It's really translational. But I think if he was sat here today, I think it's something he'd like to hear about. So there's going to be some significant overlaps from the talk that Sabine's presented. But I'm just going to reaffirm some key points. So we know that MND is defined by the loss of nerves that connect the brain from the muscle, which transmit electrical impulses. But importantly, you can think about the disease as a loss of the electrical connection between the cortical circuits and the lower motor neurons and the muscle. Importantly, these electrical changes can be detected in patients, and they change throughout the disease. For example, clinical neurophysiological assays provide key readouts of the disease stage, including forming part of the diagnosis and also monitoring progression. And some examples of those are EEG, which look into cortical circuits, transcranial magnetic stimulation, which look at the upper spinal tracts and cortical circuits, and furthermore, peripheral nerve stimulation that looks into lower motor neuron function. However, while the disease is defined by a loss of electrical connection very early on in disease, it's almost the opposite happens. It's defined by hyper-electrical function or hyperexcitability. And importantly, as Sabine's introduced, in MND circuits, what we have is a tip-up of this excitatory balance towards more excitation or hyperexcitability. So, just to reaffirm what Sabine said, why is it important? Well, it can give rise to fasciculations, other symptoms, and we know that it contributes to motor neuron injury and loss in lab models. So we know, therefore, it's a key driver of disease. So therefore, we think hyperexcitability is an important underexplored therapeutic target. And importantly, what I'm going to focus on are these cortical circuits here. Importantly, these cortical circuits are hyperexcitable long before any neurodegeneration and long before any of the lower motor neuron dysfunction that goes on. So they're a good thing to study. TMS has been introduced already, but you can measure cortical hyperexcitability in MND using this technique. And what this technique does is key. It uses pulses of stimulation, of magnetism, which induces the cortical networks to activate. The cortical networks then transmit electrical impulse down the motor nerves, which is then recorded in the muscle. I'm not going to go into it in great detail, but effectively what this technique shows is that the amount of stimulation needed to activate motor circuits, the threshold, is much lower in MND. And you can see that here in this specific bar graph. You can see that here. So in MND patients, what you get is a much lower threshold, and therefore they're hyperexcitable. So MND circuits are easier to activate. So I'm going to boil down the cortex into two different neuron types: excitatory, which drives network activity, and inhibitory, which puts the brake on. Importantly, in MND, we know that the inhibitory brake is less effective by using this technique. So importantly, a loss of dysfunction is a key feature of MND circuits, but importantly, there are no therapeutics to target this. And the goal of an aspect of the lab over the last few years is to try and develop a pipeline of lab assays that will allow the discovery of new therapeutics to rescue this inhibitory function. So how do we go about this? So number one, we require a model that's going to replicate the complex nature of the human cortex. So therefore, we've gone to the lab mouse. We want a model that replicates many aspects of MND. So therefore, we've turned to the SOD1 mouse, which has already been introduced today, but essentially gives profound MND-like symptoms. And furthermore, we also already stand on a platform where we know that there's already inhibitory problems in this model. Sabine's already introduced her work, but one key piece of evidence is that genetic manipulation of these inhibitory neurons can extend survival in this particular mouse. However, what we don't know is how inhibitory dysfunction specifically impacts networks, and as we've indicated, there's no therapeutics to target this. So we have a hypothesis that these mice display network hyperexcitability, which is driven by a loss of inhibition, and these can be targeted by new therapeutics. So to look at cortical circuits, we've looked at the mouse and we've looked at in vivo circuits. So what we've done here is used a multi-electrode probe and inserted that into the cortex. Importantly, we've done this experiment on P60 age mice, which is at the pre-symptomatic phase. To stimulate the cortex, which is needed in this particular paradigm, we've used mechanical whisker stimulation. Of course, we can't use transcranial magnetic stimulation because the because the brain of the animal is so small and we can't use that focally to activate those circuits. So when we use the whisker stimulation, it causes activation of the cortical circuits. And what you can see is there's 10 stimulations in quick fire. And what you get is an increase in the stimulation, and then what it does, it depresses over time. The inhibitory brake comes on. What happens now in SOD1? You'll notice two things. Number one, that the amplitude is much larger. And number two is that the amplitudes here don't depress back down. The inhibitory brake is effectively lost. And what you can do by measuring these amplitudes is you see that it flatlines, and you can see the depression here in the field potential. Importantly, we've done this work, this PhD student who's now passed out, Marilyn has done all this work, but effectively we've done this at multiple time points. Importantly, network inhibition persists throughout all the life course of this animal, but it only gets stronger as time goes on. Now, in vivo work is great, but anyone who's done it, it's time consuming, it's laborious, and very attritional. So we wanted to get down into a system which is going to allow us to look at drugs which will correct inhibitory problems. So what we've then done is turn to in vitro assays. So using simple primary cultures of disassociated embryonic mouse brain, we've then plated these onto multi-electrode arrays. So multi-electrode arrays are consistent with the in vivo. Those were in 3D, but this is now in 2D. Importantly, we don't need to wait 60 days to do the assay. These are highly reproducible when it comes to the stimulation. We can keep these in culture over time, so we can see how dysfunction progresses over time, and importantly, allows us access to be able to try multiple compounds. All in all, it's much higher throughput. So what you can see here is 1 electrode's worth of trace, and what you can see, the stimulation goes in at time point 0. You then see an intense amount of spiking activity, which is almost done by half the recording. When you wash in blockers of inhibitory transmission, picrotoxin, what you do is release the brake. You stimulate, and then all of a sudden, to use a scientific term, it goes crazy. Okay, you get loads of crazy spikes going on. Now, in SOD1, what you see is something very different. You can see that the stimulation, the amplitude is much higher of the spikes, and also that the spikes persist. And when you wash on the PTX, actually you don't see much going on at all. It's very good proof that there's loss of inhibitory transmission. So what we're doing here is being able to model this in vivo stimulation circuit in vitro. So, just as a brief summary so far, so we had a hypothesis that this particular mouse model manifested early network cortical hyperexcitability, and that was due to a loss of inhibitory function. We've used in vitro and in vivo multi-electrode electrophysiology, which allows us to mimic what goes on in the TMS experiment. But of course, what we're interested in is trying to get some therapeutics. I'm going to cut a long story short. We've looked at 2 broad rescue strategies. Number 1, we've benchmarked against riluzole. Effectively, I'm not going to show the data, happy to later, but effectively this has a very poor pharmacological profile for correcting network activity. So we've looked at correcting inhibitory dysfunction. We've looked at boosting the inhibitory transmitter receptors, the GABA-A receptors, which respond to these inhibitory transmission signals. Effectively, or ineffectively, they don't work. So we've then gone and looked at boosting inhibitory neuron properties specifically. Sorry, this is a little wordy, but it allows me to get to the point quickly. So the data are highly consistent with a loss of inhibitory function. We know that this isn't due to a loss of inhibitory neurons. Sabine has mentioned that already, that these neurons are in situ. They're just not performing well enough. We've also performed patch clamp recordings of these inhibitory neurons, which indicate a loss of function in these animals. And I'll come back to that a little later in the talk. So we need a way to selectively target these specific interneurons. And again, to cut a long story short, we've nestled down on a specific ion channel. Ion channels are critical for mediating the excitability of neurons. And one particular ion channel, Kv3.1, is selectively expressed by inhibitory interneurons. And it's absolutely critical for mediating this fast spiking behavior, which allows it to mediate the inhibitory response. Moreover, these particular compounds that we're working with are now being investigated in early experimental studies to restore fast spiking inhibitory neuron function in schizophrenia patients. So let's go back to our assays. And this is the in vitro assay I showed with cortical, associated cortical neurons. Here's the wild type. Here's the SOD1 going crazy. And now when we wash on a Kv3 modulator, you can see all the spikes have now been shunted back up in the right way. What happens in vivo when we take this compound back? This is preliminary work, and this has been analyzed on the train on the way up. But essentially, what you get is a nice preliminary result, which shows that these particular Kv3 modulators basically reinstall this inhibitory brake. So importantly, we think that we've got a compound that attenuates network problems effectively by rebalancing excitability. So this is where we are, and there's a couple more slides left. But many of you think, well, all of this is in mice. Well, is there a human relevance to this? And furthermore, this is in a very rare causal gene to ALS. So what about other genes? So I'm not going to go through the data here, but we've done exactly the same for these particular mouse models. And in terms of humans, what we've now done is looked at iPSC-derived GABAergic inhibitory neurons directly. So it's not been introduced today, but what we can do is take stem cells from patients and using established protocols, generate enriched populations of GABAergic neurons. So we've been able to generate from healthy and MND patients. I'm just showing one of each for clarity. We've then performed patch clamp electrophysiology, which allows us to look at the single neuron excitability of these inhibitory neurons. And you can see in the healthy neurons, these healthy, nice and fire, quite nice and spiky. And then with the C9, you can see that there's an acute loss of function. Now, when we wash on our modulator, we can see that the excitability is now largely restored. So for those wondering how all this is potentially happening, well, we've looked into it a little bit more, and I've got a bit of data here to show that. So importantly, when you look at these spikes in a lot of detail, you can see that the healthy spikes are nice and sharp. It's just great because when an action potential goes down a neuron, it needs to be precise to release the inhibitory transmission signals. In C9, you can see that it's not so spiky, it's actually quite stumpy. Importantly, we know that action potential initiation starts at the axon initial segment in all neurons. And importantly, what we've done is then looked at the axon initial segment, which is demarcated by a protein called anchorin G. Here you can see in the healthy that the anchorin G stain is nice and long. However, in the C9 and overall, you can see that it's reduced, therefore potentially explaining why these C9 spikes are stumpy. So what's this got to do with Kv3? Well, Kv3 is localized to the axon initial segment by this motor protein KIF5, mutations of which give rise to ALS. And furthermore, it binds to anchorin G. So Kv3 is localized to the axon initial segment. And we think that there's a potential disruption of Kv3 at this particular point in the neuron to cause the overall dysfunction. So again, just a brief summary. We've got our animal model where we see loss of function and we can rescue it. We've now got a human model and we can rescue it with a Kv3 modulator. So the second and last, or the last question is, how is inhibitory dysfunction relevant to all MND patients? So I'm just going to summarize a project that's ongoing at the moment. So importantly, I introduced TMS data like this. Now, compared to non-MND, it's significant, it's highly significant, and there's hyperexcitability across the population. But when you look at that population in terms of individual patients, you see there's a big spread. And in fact, everything under 0 is technically hyperexcitable, but actually everything above it is hypoexcitable. So it's not applicable to all the patients. So what we're trying to do is trying to understand this, and to try and capture that for the first time, we're trying to align MND patient electrophysiology with that of human electrophysiology. So James, Alex, a clinical neurophysiologist, performs all these assays, and then for those really interesting patients, we're now reprogramming those iPSCs. We'll make those iPSCs available once they're reprogrammed for all the community to work with. But especially in my lab, what we're interested in is putting these onto high throughput MEA systems so that we can set up a higher throughput screening set of assays. So importantly, the summary, the final slide. And I suppose it's not really a summary. I suppose it's hopes and dreams and where is all of this going. So hopefully, maybe in a few years, patients will come into the clinic and they will be diagnosed and they will undergo TMS. And maybe that they'll be separated out based on their neurophysiological profile. Importantly, we've got preclinical workup to indicate that these hyperexcitable patients might be best treated with compounds that target inhibitory dysfunction. And before we get to a clinical trial, what we need to do is an experimental medicine study, and we're in talks to try and get something down in terms of demonstrate how these Kv3 modulators improve SOD1 survival, and furthermore, how do these Kv3 modulators rescue patient inhibition problems. But all in the meantime, we're interested in stratifying these and understanding how these individual patients give rise to these different properties. And from there, we'll be able to hopefully work out new stratified therapeutic opportunities. So all of this is done by a whole bunch of people in Sheffield. They also have connections still in Edinburgh, it's great. Merck, Sharp and Dom have been involved, and also my old mentor in Dundee, Jerry, who's been a great source of pharmacology for inhibitory transmission. On that note, thank you very much, and I'd just like to thank all the patients, because there's a lot of patient involvement in this work too. Thank you very much.
Thanks, Matt. Really interesting stuff and well to time too. So Siddharthan will be happy with that. Do we have some questions in the audience? Good. One here.
Hello, Matt. Thank you very much for the great talk. My question relates to the C9 IPC cultures you were talking about. Mm-hmm. And I was just, first of all, wondering whether you saw overall percentage of GABA positive IPC differences in C9 versus the healthy control. So what was like the overall percentage in those cultures? Okay. Normally you would see higher percentage of excitable neurons rather than inhibitory.
So in those cultures, these are specifically enriched for inhibitory neurons. So you get about 90% GABAergic neurons. Okay. There's some other stuff in there, but 9 times out of 10, you're looking at an inhibitory neuron, of which they're a mixture of SST and parvalbumin. The ones that you've that have been presented there are parvalbumin, which are the fast spiking interneurons.
Okay, thank you very much. Just a small follow-up question, if that's okay. Did you also see any changes in the Kv3 in the cultures, so the ion channel that you were talking about? Did you look into any changes in the Kv3 ion channels?
So we don't see any biochemical changes in expression. So we think it's a localization problem. So this idea that the axonal transport is a big problem in ALS, we think that the KV3 ion channels aren't getting to the AIS for whatever reason. So we think that's so there's no difference on a Western or a PCR, it's being processed properly, but it's not getting to the right place.
Okay, thank you very much.
Okay. Brian, just behind. Actually, while Brian's I'm going to jump in and ask a question, which is, it was interesting, the actual initial segment linked with the KV3 channels. Did you also look at the AIS itself? Because I think there's work from Claire Meehan and Copenhagen and others, in motor neurons at least, has seen changes in that structure, which I wonder if that might also...
Yeah, so it looks like these interneurons are possibly non-plastic. You know, neurons can adapt their excitability, and the way they do that is by shifting their axon initial segment either in distance away from the cell body or shortening. So we think that there's possibly a loss of plasticity associated with those neurons, and they can't function as they should be.
Okay, good.
In that sense.
Brian?
So it's quite a broad question. Do you think your in vitro microarray studies point to a neurodevelopmental component to ALS?
Yeah, so there's multiple evidence, including the human cultures as well, where you could look at it as a neurodevelopmental impairment. However, I think probably the best way to address that is, if you look at many of the developmental properties in the human C9 neurons, for example, the resting membrane potential, the capacitance, other properties like that, they aren't changed at all. Okay, so they would be expected to lag behind as well. So it has elements of a neurodevelopmental disorder, but it's not quite there.
Okay. Question on the back there.
Hi, Matt.
Hello,
do you think there's an astrocytic component to this as well? Because if, for example, if we would co-culture the neurons with normal astrocytes, would that sort of rescue the function? Because KV3 is also expressed in the astrocytes.
Yes, we haven't done those experiments, but we do think there is a oligodendrocyte problem. Okay, so in the Brett Schneider paper, I've seen someone present it earlier, the earliest TDP-43 pathology that's evidenced in patients is in oligodendrocytes that wrap around parvalbumin interneurons in the cortex.
Okay. One right at the back there.
Yeah, great talk. Apologies if I missed it, but could you talk a little bit more about the pharmacology or mode of action of the KV modulators?
Yeah, so effectively, it's a potentiator. Okay, so KV3 acts by bringing down the repolarization phase in a fast way, so that basically the excitability repolarizes quickly, so that it can basically then depolarize a short time after. So therefore you get fast spiking. So KV3 potentiators enhance the function of that particular ion channel of that function. Is that okay?
Okay, maybe one last one. Yep.
Thanks for the talk. I was just wondering if age somehow plays into how the inhibitory circuits or cells work just in general, and that could be a sort of tipping point process for why the disease would start then.
So you're asking if there's an age-related decrease in, yes, in general. So in aging, in general, you do see a loss of fast spiking inhibitory neurons, but in MND, the rate is so much more quicker over time. It's hard to tease out exactly what is the tipping point, right?
Okay, thanks again, Matt. So we're now going to move on to a series of flash talks, so 5-minute talks from early career researchers. I think originally we would have them all up on the stage, but I think I've decided we'll just bring out one by one so you don't have to sit there and be stared at for a while. So the first of our speakers is Alyssa Corbett. Alyssa is a Euan MacDonald Centre funded PhD student who actually I've had the pleasure of working with. So I will hand straight to Alyssa and thank you.
Hi everyone, thank you, and thank you for the opportunity to present. So, similar topic, I'm going to be discussing how the communication between neurons and astrocytes, now coming into the story, is dysfunctional in pre-symptomatic SOD1 mice. So, we know that astrocytes are key cells that help our central nervous system, and they have many roles, but one of their important roles is to provide inhibition to neurons. Now, this ability to hit the brake on neuronal circuits is very important, and it can help prevent too much excitation. We know in ALS that hyperexcitability and eventual excitotoxicity are hallmarks of the disease. So this made us question, is there potential that astrocytes are losing this ability to provide inhibitory modulation to neurons? Now, importantly, I investigated this in very young mice. So this is the SOD1 G93A model. They're not going to get symptoms until around 3 months of age. All the work I'm going to show you are in just 7 to 10 day old SOD1 mice. So for these experiments, I isolated the spinal cords from these mice, and I then attached an electrode onto one of the lumbar ventral roots, and I evoked the locomotor central pattern generator, and that provides a recording as shown here. Every burst you're seeing is a burst of motor network output. Now, strikingly, the first thing we see is that these SOD1 mice at just 7 to 10 days old have hyperactivity in their spinal circuits. So we see this increase in bursting frequency, this faster rhythm, which suggests hyperactivity. Again, this makes us question, is there then potential that astrocytes are losing their ability to provide inhibition, and could that be contributing to this hyperexcitability that we're seeing? To investigate this, I completed the same experiments, but this time I tried stimulating astrocytes to see if I could prompt this inhibition from them and see if it could help calm things down. Here you see a wild-type trace. In the middle phase, TFLR, that's where I'm stimulating astrocytes and trying to provoke this inhibitory break. As you can see from this example, the wild types do indeed show a reduction in their bursting frequency. That rhythm slows down. This is good. This is the inhibitory astrocytic modulation we want to see. Now, here's an example from the SOD1 mouse. At just 7 to 10 days old, we don't really see much astrocytic inhibition when prompted. Again, across all of experiments, this holds true. So this made me think, are we just losing this function entirely from astrocytes? Can they simply not hit the brake on the circuits? To investigate that, I completed the same experiments, but this time, instead of stimulating astrocytes, I blocked their ability to provide inhibition. Here is a wild-type trace. In the middle phase, you can see the blockage of this astrocytic inhibition. We don't see much change. That might be because there's not hyperactivity of the circuit, so the astrocytes aren't really needing to hit the brake. Now, here's a striking example from one of the SOD1 mice. We see that when I inhibit the astrocyte's ability to provide this inhibition, there's severe hyperactivity of the circuit. This rhythm speeds up. This is seen across experiments. So this showed us that astrocytes can indeed provide this inhibition. We haven't lost that function, but it's actually overactive. And because I cannot stimulate it further, as shown in the last experiments, that suggests that it might essentially be a function that is maxed out, meaning that if the system was then introduced with more excitability, it might be a system that's set up for failure for excitotoxicity, again at just 7 to 10 days old. So I wanted to understand the mechanism that might be contributing to this. So all of those recordings were at the network level, recording from a ventral spinal root. Now we're moving on to the cellular level, just recording from a singular interneuron. And we see that when I stimulate astrocytes and record from an interneuron, we see in the healthy wild types that there's a hyperpolarization of those interneurons, again, the mechanism we think of this inhibition. We see that the SOD1 interneurons fail to do so. So this is our best guess at the mechanism at a cellular level that might be behind this network dysfunction. So in conclusion, what I've shown you is that in pre-symptomatic SOD1 mice, we see that there's already spinal motor network hyperexcitability, and there's dysfunction of astrocytes' ability to provide sufficient inhibition to the network and to the neurons. Now, importantly, we actually know the specific pathway and mechanism in which astrocytes usually provide this inhibition, and I further characterized where this dysfunction seems to be occurring in the circuit. So if this is something you're interested in hearing more about, I have a poster today, number 1, from 1:40 to 2:10. Would love to talk to you more about it. So thank you so much again for the opportunity to speak, for Euan and for his family for supporting this research and allowing it to happen.
Let's go turn myself on. Questions? Well, just time for a couple of quick questions for Alyssa, if there are any.
Hi, great talk. I was just wondering what you stimulated the astrocytes with.
Yeah, it's a good question. So it's a modulator called TFLR. TFLR is an agonist for a receptor called PAR1, and PAR1 is, we've seen, selective to astrocytes in the spinal cord. As a control, it did some immunolabeling, and PAR1 expression is unchanged in the disease.
Thanks.
I'll just show. Yep. Are you able to measure any changes in astrocytic uptake?
That's a good question. So we haven't looked at astrocytic glutamate uptake or sort of the EAT2 side of this yet. That's something that would definitely be interesting. We are moving on to astrocytic calcium imaging. So not quite the glutamate side, but it will still be interesting to see if their activity is different. That's a good question.
Okay, one last one.
Hi, there's different populations of astrocytes. Do you think they're maybe involved in the sort of protective areas, and this might be because of the differences in the astrocyte populations?
That's a really good question. We haven't looked into that. That would be very interesting to see if there are different properties of the astrocytes that might be responding differently. Potentially that's something after calcium imaging that we could look into, but I'm not sure.
Okay, great. Thanks very much, Alyssa. Okay, so our next rapid talk is going to be from Hannah Crick, who is another Euan MacDonald Centre funded PhD student studying in Edinburgh in Tom Gillingwater's lab, and another person I know well because I remember her doing an honours project some years ago in St Andrews with us. So good to see Hannah back. So looking forward to her talk.
Thank you, and thank you for having me to talk about our new mouse model. So I work on UBA1-mediated motor neuron disease, which is otherwise known as X-linked spinal muscular atrophy, which you might not have heard of because it's very rare. In its most severe form, it's a congenital onset disease featuring reduced muscle tone, a lack of reflexes, and congenital joint contractures and fractures. And it's highly severe and usually causes death by the age of 2 years old due to respiratory failure. However, with the broadening use of diagnostic sequencing, we are seeing that there are actually a range of atypical cases that are coming through. For example, this is George. He is 6 years old and living with X-linked SMA. He was diagnosed in infancy and is unable to walk, but he's been stable throughout his childhood. And he's not alone. There are many cases coming through that, like George, and milder. Some people are diagnosed in adulthood. So we believe this disease is far more variable than the current definition allows for. So the mutations that cause X-linked SMA are in ubiquitin activating enzyme 1, which is located on the X chromosome, giving the disease its name. And all the mutations represented by the colored lines here cluster in exon 15. So this gives us a very clear genetic cause for the disease. But despite this and the mysteries around the massive variability in it, there's been little research into it, and there is no current treatment for this devastating condition. So my PhD has been focused on characterizing a novel mouse model for X-linked SMA. This was made in collaboration with the Mary Lyon Centre at MRC Harwell, using CRISPR-Cas9 to reproduce a human single base mutation. This was one that was well characterized in a young boy who died in infancy from X-linked SMA. Our mice, however, have bred well, they survived gestation and infancy, and we have characterized them into adulthood. So the mutant mice here you can see are the black dots, and they are averaged out in the blue line compared to their wild-type litter mates. So throughout adulthood and lifetime, we see a 10% on average body weight loss in the mutant mice. Using echo MRI to look at body composition, we saw this loss was specifically due to a difference in lean mass. And ruling out for body size and organ changes, we identified this specifically as difference in muscle mass. At the same time, at 12 weeks old, we see this translates into a reduction in strength as measured in the grip test. And at the cellular level, at the same time point, we saw changes at the neuromuscular junction. So in green here, you've got neuromuscular filaments coming in and presynaptic vesicles highlighted. And then in magenta, you've got the muscle side of the NMJ and the acetylcholine receptors labeled. So we've seen the mutants that there are smaller presynaptic terminals, which is indicative of pathology at the motor neuron side. And indeed, at 6 months of age, we see a difference in the number of spinal motor neurons. So we're losing motor neurons in the mutants. It's about 25% loss. Gathering all these features together, we have a new model for motor neuron disease. And as far as we are aware, this is the first mammalian model for motor neuron disease mediated by UBA1 pathology. So in identifying these pathological features of the model, we've been able to start looking at a gene replacement therapy for X-linked SMA. At the same time, this has allowed us to look at the mechanisms of the disease, so specifically looking at changes in ubiquitination and UBA1 activity, but also changes in UBA1 isoform expression and why this might be so important for the central nervous system specifically. We can also look at the different disease-causing mutations in the structure of UBA1 and how they may be relating to the variability in X-linked SMA, as long as with a number of other factors like other MND-causing mutations that may be affecting their presentation. So, and this might help us work out why cases like George's, which we believe our model represents more closely, differ so much from the cases that were initially used to define this disease. Thank you very much, and thank you to Euan MacDonald Centre for my funding and for your listening today.
Thanks very much, Hannah. Do we have a couple of questions for Hannah? Oh, yep, just here. Another one at the back. Yep.
Nice
Sorry.
talk, thank you.
Oh.
It looked, sorry.
All right, off you go, Richard.
Okay, thanks. Thanks, very, very nice talk. Just a quick question. It looked like from your NMJ morphology that in addition to the area of the nerve terminal being reduced or the end plates being reduced, that the pre-terminal axons appeared thicker to me. Is that something that's consistent?
We didn't see a change in the axon width across them, but it's something we'll be doing many more levels of NMJ morphs, so we can definitely look at that in the treatments as well.
Sorry, the mutation, are you mutating the mouse UBA1 or are you introducing a human mutation?
So we've just introduced a single base change in the mouse, but there is very high homology between the human and mouse gene in UBA1. It's a highly conserved gene.
Okay, thank you very much, Hannah. Okay, so our next speaker is Dr. Johnny Tam, who's an MRC Clinical PhD Fellow here in Edinburgh, supervised by Savanka Pal, Siddharthan, Chandran, and Oliver Watts. So when you're ready, Johnny, thank you.
Hello, everyone. My name is Johnny Tam, and I'm an early career neurologist and a final year PhD student at the Euan MacDonald Centre. And I'm supervised by Professor Pal, Professor Chandran, and Dr. Oliver Watts from Speak Unique, which you'll hear more about after lunch. And I have the honor to present some of the work of our research group looking at digital speech biomarkers of dysarthria in MND. And this collaborative project is only possible through Euan's interest and his legacy in both speech science and MND research. So we know that dysarthria, or speech slurring, is a prominent feature in MND. But the way that we assess it in clinic at the moment is very subjective and is qualitative. The most common way we do that is through the speech question from the ALS functional rating scale, which is on the screen. It's an ordinal level of 5 descriptions that we provide through subjective evaluation of someone's speech intelligibility. So it's hard to standardize, and it also requires, a trained ear specialist to make these subjective assessments. So there's massive potential here for us to develop digital speech biomarkers, which can potentially be objective, be quantifiable, and we can actually standardize these assessments better. We can utilize common and available technologies that are available to our patients to provide low burden and brief assessments that are not only more ecologically valid, but can also potentially decentralize assessing our patients and monitoring them in the community. So to achieve this, we gave ourselves 3 broad aims. We firstly wanted to establish clinically annotated neurological speech data sets in these conditions, and use these speech recordings to conduct acoustic analysis with machine learning models and evaluate them in a clinical context. And we want to do all of this in co-production with our patients and our caregivers, because any assessment methods we develop has to be acceptable to them as well. So the first thing we did was to develop an app, as you do, with speech recording tasks in them. And we did this alongside our patients and our caregivers using the feedback they provided for us. And we included some reading tasks and some short cognitive tasks in order to elicit the speech, which we deployed in some observational studies. The app takes about 10 to 15 minutes to complete, and we give the opportunity for people to do this in clinic or really anywhere in the world at home. And Bruce, who's one of our participants, just told me during a coffee break, he did this all the way in Australia, which was very impressive. We've been doing this for about 3 years now, and we've collected data from over 800 participants, including over 100 people living with motor neuron disease. And crucially, these recordings are accompanied by detailed clinical information that we can use to train our models. So the first step is to leverage machine learning speech models to look at how good they are actually identifying disease. And you can see here a summary of the diagnostic performance of one of these models classifying controls versus people who have motor neuron disease. But that's just the first step. Rather than just detecting the speech disturbance, we actually want to monitor its severity. So based off of initial work led by Speak Unique, we devised a novel semi-supervised learning scheme. Essentially, we take and we organize our data sets into triplets. So for each sample in our data set, we find a similar sample or a dissimilar sample based upon the ALS Functional Rating Scale speech score. And if you do this, you can create millions of these triplets. And if you get a model to train on these millions of triplets, it allows the model to learn for itself the differences between these recordings. And what we found was that when we then assess this on our data sets, you can see here in the top two plots, it can discriminate diagnosis quite well, but it can also correlate to the ALS-FRS speech score. And it's very excitingly for us, when we take these models trained on our English-speaking Morningside accent participants and apply it in data sets from a completely different language. So here in Taiwanese Mandarin, we can produce the same pattern and correlation as well. This just gives you an example of what that looks like at an individual level. So on your left is a speech score in blue from a control participant who's consistently high. On your right in blue is the speech score from someone living with motor neuron disease, and you can see that decreasing gradually over time. And in orange, superimposed, is their ALS Functional Rating Scale speech score. So you can see instead of a stepwise decline, we can capture that decline much more sensitively. So in summary, we can assess our participants using easy and remote monitoring of dysarthria severity. We can use machine learning models in order to assess this severity of dysarthria with validity across languages. And longitudinal data sets that we're developing will help us develop further evidence for these predictions as biomarkers. Thank you for your attention. Thank you for the clinicians, our participants, and again, thank you to Euan and his family for enabling this work.
Great, thank you, Johnny. Do we have questions for Johnny? Yep, being at the front. Maybe yell and like it. Or we'll get the mic.
That's super nice. So if I got it correctly, you do not have to train a new model for a different language, but you have a universal model that applies to literally all the languages that exist. Is that true?
That's what we hope to prove.
That's what you hope. Okay. So then comes the question. Do you know, or can you extract the parameters that define the score the most, that drive the score the most? I'm pretty sure you have access to all of the different. Is it speed or the breathing in between, or what is it that tells them that this is an ALS patient?
So that's one of the limitations of this deep learning type approach. It is a black box, but we're hoping to doing some ablation studies in order to interrogate actually what about the speech signal these models are predicting on. So that's work to be done.
I think we had a quick question, which is, I think it's really interesting that you've got, the cross-cultural different languages showing up. I wonder if there's any power in that as well in terms of the differences between the languages and what you pick up. And I'm thinking, for example, of Mandarin speaking versus English, and I know that Mandarin's got this melodic tonal element, which even uses different parts of the brain. Do you think there's anything that you can utilize amongst the sort of differences between the languages to gain insight? It's probably a crazy question.
I think we need to find out more about the differences that the model is predicting on between these languages before we can answer that. But the common example I give is speaking rate, which is used in the MND ALS context. It's much higher in languages like Spanish. So how do you control for that?
Right.
And this is potentially one way to bypass those confoundings. Yeah.
Okay, great. Good. Okay, thank you very much. Okay, the last of our flash talks is Áine Heffernan, who's a postdoctoral research assistant in Edinburgh working in Siddharthan's lab. Thanks, Áine.
So yeah, hi everyone, I'm Áine and I'm a postdoc researcher in Professor Chandran's lab based here at the University of Edinburgh. And today I'm going to be talking to you about my work relating to alterations in the oligodendrocyte lineage in ALS. My primary research interest lies in understanding neuroglial crosstalk in neurodegenerative conditions. As this is in recent years, a lot of evidence has accumulated which has shown that dysregulation in glial cells like microglia or astrocytes can induce neuronal dysregulation. In contrast, and notwithstanding that this is a bidirectional communication, the influence that dysfunctional neurons can exert on glial cells is comparatively understudied. So therefore, I was interested in understanding the effect that neuronal pathology can have on glial cells. And in particular, I focused on the effect of neuronal cytoplasmic TDP-43 aggregation, as this is a hallmark feature of ALS, present in 97% of ALS cases. So to explore this, we used the RNLS8 mouse model, as this has neuronal TDP-43 pathology just in the neurons. We then used FACS to isolate different glial cells, so astrocytes, microglia, oligodendrocytes, and endothelial cells from this mouse at an early disease stage where the motor phenotype has begun, but prior to gross motor neuron degeneration. We then carried out unbiased proteomics in collaboration with Raj's group at the University of Dundee to investigate the molecular changes that could be experienced by these different cell types. As expected, we saw increased expression in inflammatory-associated markers across all the cell types, but we saw some interesting results in relation to the oligodendrocytes. When we looked at the different expression of proteins associated with different lineages of the different parts of the oligodendrocyte lineage, we noted that there was an increased expression in proteins associated with this pre-myelinating oligodendrocytes, so increased expression of SOX10 and MYRF, while we saw this decreased expression of myelinating proteins such as MOG and MBP. When we looked at a later disease time point, we could still see that there was this increase in CNP and MYRF, but now that we could also see this increase in MOG myelinating expression. And we think that this may represent a compensatory response, as this has been reported in some other neurodegenerative models. Next, we wanted to explore whether this finding had any human relevance. And to do this, we performed weighted gene co-expression network analysis on bulk RNA sequencing data generated by the New York Genome Center, which has around 85 ALS cases. With this, we identified different networks of dysregulated genes and noted that some of these altered networks, such as the light cyan network shown here, demonstrated increased expression of genes associated with oligodendrocyte maturation, such as MYRF, which we had seen in our mouse model. Interestingly, then, when we related these networks back to some clinical phenotypes, we noted that this was only observed in people who had a bulbar onset form of ALS, highlighting the heterogeneity that can exist in people with MND. We then further validated these findings by immunohistochemistry in an independent cohort from here in the Edinburgh Brain Bank, And in the BA4 motor cortex, again, we could see that there was specifically an increase in MYORF positive cells in people who have a bulbar onset form of the disease as opposed to a limb onset. So in conclusion, we observed that neuronal TDP-43 pathology can induce alteration to oligodendrocyte lineage dynamics in this RNLS8 mouse model, and that increased expression of genes associated with oligodendrocyte maturation is also seen in people with MND, but specifically those who have a bulbar onset, highlighting the disease heterogeneity. Future work in the lab aims to build on these findings by firstly exploring in further detail this relationship between TDP-43 and the oligodendrocyte lineage in human stem cell models, which is work led by Sophie, and in zebrafish models, which is work by Marcus. And we're also interested in continuing to explore the disease heterogeneity in people with MND in human postmortem tissue, which is work being led by Jade. So with that, I'd like to thank everybody in the Chandran lab in Edinburgh, and also in the groups in Dundee, and the people who donated their tissue for this project.
Thank you much, Áine. Do we have some questions?
Hi. In your proteomic slide, you had a big cluster of endothelial cells.
Yep.
Did you ever look at those anymore at all, particularly in the early and late onset patients?
Oh, we didn't look in patients. We just looked in the mouse for the endothelial.
Did you do anything else for those endothelial cells?
No.
I would have liked you to have done.
Yeah.
Great talk. Do you find any evidence for TDP dysfunction in your oligodendrocytes, despite the fact that in this model you do not express the delta NLS in oligodendrocytes?
I haven't seen anything in this model that would indicate that.
So no nuclear depletion, mislocalization, no nothing?
No, not that we've seen. But
okay, thank you.
Okay, so that was the last of our flash talks. So we've got one more talk to take us to the lunch break. So this will be delivered by Owen Gwydion James, who's a Wellcome Early Career Research Fellow at the UK DRI in King's College London,
Yes.
and is another one who has been here previously and is known well to us in the centre because he did his PhD in Edinburgh. So welcome back, and we look forward to your talk.
Thanks. Yeah, similarly to Matt, it really does feel like coming home. It's a privilege to be here. I did my PhD in Siddharthan's lab, and this meant that, well, it was partly funded by the EMC, and I had the privilege of meeting Euan and his family on many occasions and interacting with patients and their families, which was a wonderful thing for a basic scientist because it really adds that personal aspect when you see the impact of the disease firsthand. And I had a great time in the centre, getting involved in different fundraising activities like the annual Relay Marathons and the golf fundraisers and patient scientist evenings and events like this one where we got to interact with the different EMC researchers across Scotland. And I also had a good time scientifically. It had its challenges, but I was tasked with my PhD to develop a human stem cell-derived in vitro model of myelination, which at the time was kind of inaccessible to us. And the turning point really came when we applied an organoid approach to this problem, which allows for much more physiologically relevant cell-to-cell interactions. And we were able to generate systems that gave widespread, compact myelin development with proper nodal and paranodal organization, and then applied the system to look at neurodevelopmental and degenerative diseases like ALS, as well as asking kind of fundamental questions around human myelin biology. And so it's really nice to hear kind of mention of oligodendrocytes being increasingly implicated in ALS, and I hope some of this research can be applied to this kind of model. After my PhD, I really wanted to take a deeper molecular dive into disease modeling, and so I joined the lab of Yone Ule at King's College London, who's the head of the DRI Centre there. And the goal was to kind of bring stem cell modeling and together with his expertise in looking at RNA and protein interactions in health and disease. And currently I'm about a year and a half into an early career award from the Wellcome Trust to look at the role of genetic modifiers in ALS. So much of what I'm going to tell you today is quite preliminary. I'm going to tell you about some of the models that I'm working with in the lab and setting up, and kind of the results that we have so far and where we're going. I think it'd be worth just to talk a little bit about RNA and protein complexes, because this might not be everyone's background. RNA is decorated by proteins in the cell, and these RNA protein complexes can exist diffusely in the cell, or they can come together and then phase transition into liquid-like droplets. And these liquid-like condensates are essentially membraneless organelles that have very important specific functions all throughout the neuron. They regulate functions like RNA splicing, RNA transport, where the RNAs need to be kind of packaged up and taken to different parts of the cell, and stress response as well, and stress granules. And importantly, some proteins have a higher propensity than others to demix in this liquid-liquid phase separation. And their overall kind of dosage in the cell needs to be really tightly regulated, because increasing protein levels can allow inappropriate demixing and the formation of solid-like condensates, which are the seeds for fibrils and aggregates that we see in disease. When it comes to ALS and FTD, the link to RNA dysregulation is clear. We know that if you mutate a number of different RNA binding proteins, you actually can, this is a causative mutation for the disease. And also at end-stage disease, we see the misaccumulation of RNA binding proteins like TDP-43, which is normally present in the nucleus, get misaccumulated into cytoplasmic inclusions. The problem that we face, though, is that of disease heterogeneity. So each of these proteins is going to regulate different RNA processing events. And what we're trying to pick apart is potentially the idea of convergent pathways, and looking at how and whether or not there are common dysregulation events underlying these different pathways. For me personally, something that I'm really interested in is this idea of disease induction and what happens during disease induction. Because even in the presence of a familial gene mutation, the disease still takes 50 to 60 years to manifest. So for me, this really tells me that there are processes that are happening early on where cells are able to tolerate genetic stress and potentially upregulate adaptive pathways that allow you to buffer that genetic stress. And so I'm interested in asking kind of what are the direct consequences to disease induction, and what are the pathways that are being upregulated in response, and of which eventually fail with increasing age or too much stress that ultimately leads to disease. And one way we can modulate this process is by looking at genetic modifiers of disease. So these exist for ALS and FTD, but are poorly understood. So there are examples in the literature where if you take a transgenic animal model that demonstrates ALS and FTD phenotype, and if you additionally knock down and select RNA binding proteins, you can actually rescue the disease. And you can rescue, for example, phospho-TDP-43 levels, and the number of chat motor neurons. But how and where do genetic modifiers work? Do they work similarly across genotypes of different disease, and how they impact on RNA dysregulation is currently not known. One of these modifiers is of interest, has been a long-standing interest in the ULE lab, and this is Staufin 1 and 2, which I'm going to focus on today. So Staufin 1 and 2 are homologs, and they're kind of rare in the RNA binding protein world because they are double-stranded RNA binding proteins. So where the RNA kind of falls in on itself and forms duplexes, that's where Staufin will bind. And they're highly expressed in neurons, and it's been shown to regulate processes like mRNA localization, dendrite morphogenesis, and synaptic plasticity. And work done in the ULE lab by other members of the team has looked into exactly kind of where does Staufin bind and what kind of RNAs Staufin binds. And what you can see on the left is a circus plot, which shows you the points of contact in a single mRNA where Staufin binds. So you can see that Staufin predominantly binds to the 3' UTRs of lots of different mRNAs. And that this is especially important for neurons, which tend to have kind of the longest genes and the longest 3' UTRs. The 3' UTR really acts kind of as a postcode for where the mRNA should be shuttled to. And many of these RNAs are also locally translated. And so Staufen is really well placed in this feedback mechanism that's supposed to regulate dosage control. In neurodegeneration, Staufen is overexpressed and upregulated. And much of this work comes from Stefan Puls's lab, but we can see this in patient fibroblasts as well as in brain tissue. And his team also demonstrated that in the TDP-43 transgenic mouse, if you knock down Staufen, you rescue phospho-TDP-43 levels and chat numbers and increase the longevity of the animal. And so my interest in genetic modifiers is kind of quite broad, and we're looking to maybe discover new ones, but for the time being, we've taken Staufen1 as a candidate genetic modifier. And so what I did in the lab was I generated a Staufen1/2 double knockout iPSC line with CRISPR, and we can see that it effectively gets rid of the relevant band there for Staufen1 and Staufen2. These cells are viable, and in the lab, I used the kind of NGN2 overexpression model of neurogenesis, which allows for rapid generation of cortical ion neurons. And we don't really see any difference in the neuronal viability or the neural development. What I then did, with the help of Ella Nightingale, who's a technician that works with me, was because we know that Staufen might play a role in feedback mechanisms at the nucleus as well as in the periphery, we fractionated these cortical neurons into nuclear and cytoplasmic fractions and sent them for RNA sequencing. And in the lab, we do quite a lot of RNA sequencing, so there will be quite a lot of data on this, to try and tease apart some of the underlying changes that are happening at the transcript level. And you can see in the cytoplasmic fraction, we see Staufen2 and Staufen1 being kind of the top downregulated hits, which further validates the system. What was quite nice to see was that when we look at the differential gene expression for RNAs that are changing in the knockout, we see that processes and genes involved in the axon and the cytoskeleton and dendritic formation are all up in the Staufen knockout cell, which we think is kind of a feedback compensatory effect. And those processes that are going down in the knockout cell are all ER-associated transcripts, which again fits in with what we know about Staufen, where it's involved in regulating the processing of RNAs that are due to be, or genes that are due to be translated and shuttled to the membrane. And so now we have a good model with which we can study Staufen in neurodevelopment, and I'm interested in applying this to models of neurodegeneration. And I'm going to talk about a couple of different models that I've kind of adopted in the lab, some of which I'm developing myself, and others I've worked closely with colleagues at DRI King's. So firstly, I'm going to talk about the TDP-43 inducible depletion system. So this is a system that was developed by Mark David Roop. And it's very nice because what it does, it allows you to deplete TDP-43 at a time and place of your choosing by tagging it with a destabilizing domain. And in the absence of the stabilizing ligand, you get degradation of your protein. But if you add the ligand, then you can stabilize TDP-43 to normal levels. And what I did was then take these iPSCs, differentiate them into neurons, and looked at the changes in transcriptome by RNA sequencing 24 hours, 48 hours, and 1 week after TDP knockdown. And again, this is kind of looking at disease induction and what pathways are changing immediately. And what's nice to see is that we see the usual suspects. So all of these top candidates, top targets of TDP-43 are well known. And Staufen2 actually is the most sensitive gene that's known to be downregulated in response to TDP-43 depletion. And I'll spend a couple of slides talking about Staufen2. It's a microtubule associated protein. And in intron 1 of its gene, there is a cryptic exon, which normally isn't spliced into the final mRNA of Staufen2. It's, splicing is repressed by TDP-43. But if you lose nuclear TDP-43, you get increased amounts of mRNAs containing this cryptic exon, which leads to early termination of the transcript and reduced levels of protein. And the consequences of losing STAthmin2 are that you get a quite severe neuropathy and denervation. And what's interesting is that if you restore STAthmin2 levels in the absence of TDP-43, you can actually rescue axonal regeneration. And so whilst there are lots of things changing in response to TDP-43 knockdown, I think STAthmin2 is quite an interesting target, and I'll come back to it later on as well. The other system that I've used and moved on in the lab has been thanks to Sarah Mizielenska, who's another colleague at DRI King's, where we add synthesized PR peptides to our neuronal cultures. And so these DPR peptides are the same dipeptide repeats that are translated from the C9orf72 repeat expansion, which is the major cause of ALS and FTD that we're aware of. And a lot of work has gone into how these different DPRs lead to different types of neuronal toxicity. And what I've done is demonstrate that if you add the PR peptides to the cortical neuronal cultures, we see very nice depletion of nuclear TDP-43 content, which is dose dependent. And this is a very nice model of TDP-43 proteinopathy and end-stage disease. If we do RNA sequencing on PR-treated cells, we see the types of GO terms that we'd expect to see, so things involved in shutting down translation, immediate stress response, Parkinson's disease, and again, it gives us kind of confidence that we're seeing things that are representative of end-stage disease. And when we were doing this experiment, I was also interested in looking at how we could potentially correct some of these changes. And I decided to use mTOR inhibition as a way to do this, because it's been shown that mTOR inhibition can restore TDP-43 levels in the nucleus, as well as restoring the molecular dysfunction that's associated with this loss. And indeed, if we look at the PR alone compared to the PR plus rapamycin, we can see that a lot of the changes are dampened when you add rapamycin on top, including STAthmin2. So again, this is a nice model where we can see, have a readout of TDP-43 loss of function and restoration when we modulate what's going on. This is still kind of preliminary, but what we're looking now is comparing these two different systems, and we see lots of different changes happening, of course, that still need a bit of work to tease apart, but demonstrating the data like this, I think is quite interesting. So you can see that STAthmin2 is down in both the PR treatment as well as in the TDP-43 knockdown, and that it gets restored with rapamycin. Now, jumping back to STAUFEN, the reason why I've been talking about STAthmin2 so much is that I noticed in the data sets that STAUFEN binds all of the different STAthmins, and actually we see increased levels of STAthmin2 expression in the STAUFEN knockout cells. And so what we're seeing here are at the top iClip crosslink sites, showing that this is a selective binding of STAthmin2 by STAUFEN, and this is happening. This is happening at the 3' UTR of STAthmin2, which is exactly where we'd expect STAUFEN to bind. And interestingly, in the knockout cells, we see increased RNA coverage from RNA-seq coverage in the knockout cells compared to the control, showing that we see increases in STAthmin2 overall. And so this still needs to be kind of worked up a little bit, but I think it's a really interesting aspect, and potentially one that we can chase as to how STAUFEN is potentially acting as a modifier of the disease. We also compared the STAUFEN knockout cells to PR data sets and added PR to control and knockout cells. And whilst PR has very global effects, as you'd expect, it's a very toxic type of treatment, we also see differential responses in the STAUFEN cells in response to PR. And it's interesting that we see in the presence of STAUFEN, the major changes are kind of forebrain development and things to do with the axon, a lot of genes that STAUFEN are known to regulate. And in the absence of STAUFEN, we see different changes in TGF-beta signaling and cytokines and integrated stress response. And this is something that we're still working up now. Really, it's important because it's helping us identify the types of assays that we need to undertake in order to find those key differences in the Stauffen knockout cells. So to summarize what I've told you so far is that Stauffen is an important regulator of cytoskeletal, synaptic, membrane-bound RNAs. We can model proteinopathy in neurons using PR peptides, and this has global effects on translation across both cell lines, but potentially some interesting differential effects as well. But of course, the major question is any of this actually protective? And one of the things that I've done to look at this so far is recovery from stress, because we know that Stauffen also localizes to stress granules. And so I was interested in asking, how do cells respond to stress in the absence of Stauffen? And the way I did this was I treated my cortical neuron cultures with sodium arsenate for a couple of hours and allowed them to recover, and then looked at the number, area, size of stress granules at maximal response, and as well after a couple of different time points of recovery. And we can see that there's a trend for increased amount of total response in the Stauffen knockout cells, which is resolved faster compared to the control. So potentially something to be chased here. And this needs to be worked up, and I decided to look at this in a different way using a G3PP1 EGFP virus, which allows me to live image stress granules. And I developed this kind of repeated stress paradigm, where in the INK site we can measure stress granule formation and disassembly over a period of 24 hours, 48 hours, whatever you want. And what I did was I applied stress, allowed the cells to recover, applied it again, allowed it to recover, and applied it again. You can see an exaggerated response in stress with each bout of, exaggerated response in the number of stress granules with each bout of arsenite stress. And I was hoping to include in here the Stauffen data, but it was hot off the press and it didn't quite come in time, unfortunately. But this is something that I'm working on at the moment. So from the RNA sequencing experiments, the things that we're going to be doing next are with collaborations with Andrea Sarria's team, looking at axon guidance, axon outgrowth, and regeneration, which potentially might show assessment to specific gain of function. And as well as looking at the kind of synaptic changes with the electrophysiological recordings, either with GCAMP or with multi-electrode arrays. And so in the future, I'd really hope to tell you a bit more about Stauffen and potentially other genetic modifiers of disease. And I realize I've been actually quite quick with this talk. It's 20 minutes, but it's lunch next, so that's okay. Thank you very much for your attention, and thank you just to highlight the people that have helped me in the lab. Euan and Ala have been great, and Mark and Sarah for collaboration as well. Thank you very much.
Thank you very much, Sean, and thanks for keeping us on time. Do we have some questions? Judith, have we got a mic this way?
Thanks. You said that Stauffen binds to double-stranded RNA. Do you know if it binds to any of the repeat RNAs that you get, say, in C9orf or other diseases?
It's a good question. From what I remember, those RNAs, there's lots of charge domains in them. And so because they're quite short repeats, I don't think they necessarily form such they fold in on each other in the same way that you might get with a long 3' UTR. Most of the duplexes that we see that Stauffen binds are kind of the long, the most distal end to the most proximal end of 3' UTR. So you get these big loops that then come and bind together. And with such a short repeat expansion RNA product, I don't think it would necessarily form that kind of duplex, but this is not something that we've looked into.
Yeah, I think you get G quadruplexes in C9orf, but in conditions like DM1, you get very long extended hairpins.
Oh, that'd be really cool. Okay, yeah. Thanks.
Great. Bhuvnesh, come back.
Hi, Owen. It's a great talk. Can I ask you, Stauffen is also known as a splicing factor. Do you guys have looked at the alternative splicing in your models, and how do they compare to the TDP loss of function and the other?
Yeah, we haven't so far. So all the RNA sequencing that we've done has been 3', which is a very cost-effective method for us to just look at mRNA changes. For alternative splicing, you really need to do full-length mRNA, and bit more of a larger study. It's something that we'll probably do once we've identified the right type of stress that can maybe reveal those Stauffan dependent changes. But yeah, thanks.
In the middle here. Yeah.
Excellent work. My question is with respect to the mTOR rescue, is it specific to the C9 or the TDP-43?
Oh, that's a really good question. I don't know. I think it's specific. Actually, no, I do know. Some of the TDP-43 models have shown that rapamycin and different mTOR inhibitors can restore TDP-43. So it's not a kind of C9 specific thing, but we do. There's a very strong association between C9 off and changes, baseline changes in mTOR. So potentially a stronger association to C9, but definitely not specific.
Thank you.
Exclusive.
Okay. There's no more questions. Thank you much again.
Thank you.
So a big thanks to all our speakers for that session and for keeping us on time and for some fantastic, interesting, novel findings being presented there. Just a quick note that could you please take all of your things out of here for the lunch break, because we've got different folk joining us in the afternoon. We may need to move some things around. So importantly, there is lunch now, but also posters to be viewed as well after lunch. So enjoy both, please. Thank you.